Thank you so much. This is the warmest welcome.
Good morning.
Good morning.
While we wait for others, would anybody like to share their thoughts or feelings?
Hi, Raidu. Good morning. Vikas here.
Good morning. Good morning.
So I just thinking yesterday, about the memory and inattentiveness of humans. So generally,
And I lost after memory.
inattentiveness.
Inattentiveness. Huh?
Yeah. So while we are designing a particular UX, user experience for a person, so the memory and inattentiveness, what is the maximum consideration of these two factors that we can incorporate in our design or consider? So as design for tolerance is also one of the aspect in this, like ability of considering of a human committing error and considering that we design the UX. So what can be the some conditions, especially which are related to neuroscience, that we can take care of, especially in these factors?
So your question is, there is one on the one hand there is memory, and on the other hand there is this inattentiveness. Huh?
Yes, right.
And what is your what are you trying to do? You are trying to minimize inattentiveness versus
Memory loss.
while enhancing the memory or some.
Yeah, correct.
So one thing that readily comes to my mind is what is this? In the olden days, during festivals like Sankranti or something like, in the villages, they used to have this all night. Not Sankranti, I think. During this village deities, Amorlu, Amor Jatra.
Amor Jatra.
During that time, they have this all night Burakadar. Basically, the guy will know some bunch of people would be telling Ramayana or Mahabharata. And most of the villages, pretty much everybody knows about Ramayana and Mahabharata. And since most of them come after dinner, and they have been very busy during the daytime also, during this more jatras.
Right.
So they tend to nod off. So whenever the person who is telling Ramayana or Mahabharata the truth, what they do is while saying Ravana abducted Sita and Rama went searching for Sita, and then let's say at that point he noticed people, most of them are nodding off. Then he will say, Rama decided to send Ambani to find Sita. Then the moment they hear Ambani, everybody will be like, what the hell are you talking? Where did Ambani come into Ramayana? There is no Ambani in the Ramayana. So
Right.
these are known as fail stories or Pettakadalu. So
Burra Kadala, Pitta Kadala, something like that.
I did not get what you are saying.
Like we call as Burra Kadalo in Andhra.
Yeah, in the main Burakada, they will have this Pettakadalu.
Okay.
Pettakadalu in English, it will translate to, I think, quail stories. Quail is a small bird.
Yes,
So
yes.
the idea of this Pettakadalu is to throw in something that is completely unexpected.
Right.
In the even though they are nodding off, they're in the they will be semi-conscious. And the minute they hear Ambani, where the hell is this Ambani come from? What is he talking about? So
Right.
that will bring back the attention. So even I do that in the class. So some of that might come across as like, what does this have to do the entire the bigger narrative that he is telling about?
Right, right.
Somebody was saying something?
No, I'll go ahead, please. Nothing.
Yeah, so that is the idea. I do that once in a while, if you know. And in this particular lecture, there'll be a couple of Pettakadala or in the main Borakala to draw
Okay.
attention.
Okay.
So I don't know whether this strategy is going to work. So basically, you disrupt their expectations. So everybody has an expectation. You remember we talked about the brain. Brain
Right.
itself stop paying attention when something is going in a predictable manner and
Right.
starts to predict it. And so it shuts down the seamless processing itself, something that is coming from the outside.
Right.
And then it more or less goes on this is seeing, hearing what is expected to, what the brain expects to hear.
Right.
But once you throw something unexpected in, the brain will wake up, so to speak, and then start paying attention again. That is, I don't know how it will translate into your explicit this thing.
Yeah. I think these are very genuine factors of a human brain, that forgetfulness and inattentiveness.
Yeah.
So why can't we as designers consider these also while designing any user experience or a user interface? So this is a neurological aspect that we can address, I feel.
Yeah, there is a very good book by Daniel Schachter called Seven Sins of Memory, if I'm not mistaken. There he talks about various ways you can miss, what is that? Misremember, so to speak. False memories and various, you can even remember things that never happened to you. So Daniel Schachter, he used to be the chairman of psychology department at Harvard, and that book is he has been working on memory for a long time. It catalogs all this. You see, various ways in which you can. So. Say for example, even say even a simple, I think this is even Kahneman did some work on this. I think I forgot the title. Living and remembering the life that remembering or something. I think he might have even written a book on it. So say for example, if you ask somebody, how was your weekend? He might say, nothing much, same. He or she might say same old thing.
Right.
But if you precede that question, it say, did you go on a date? And then if you went on a date, then how was your weekend? Would be with respect to the reference you gave. So the way the same question will be answered, good or bad. You can pretty much you can get him to say, how was answer the question how his weekend was, you can elicit any answer that you want by priming it with some other question or statement. So how people remember things or forget things has a very there is a very good understanding when it comes to memory. I think Schachter's book is a very good place to start. Daniel Schachter, Schechter, Seven Sins of Memory, I think.
Yeah, Sense of Memory by Daniel Schachter.
Yeah.
Yes. Great.
Shall we wait?
Yeah. I think we have 18 students here.
Yeah.
12 more to come, I guess.
Yeah. So in the meantime, please continue sharing your ideas. Or
Yeah.
Hello, Raidu, Anshu this side.
hi.
So we were talking that day about how designing for differently abled people helps to design better products in general, right?
Yeah.
So from the perspective of neuroscience, if somebody is suffering from issues with their nerve cells and receptors and the like, are there any studies done in order to how to design better for such people?
So the front end part is very receptors and is right now you can replace the photoreceptor layer, you can replace the retina, and you can have artificial stimulation like so the front end part I don't think is that difficult. It's the thing that comes after that. You remember we were talking about when this visually differently able people, they have a difficult time most of the time. And see, right now when we look around we see a world of objects. But this world of objects is a creation of our mind or brain, so to speak, because what there is out there is basically the wavelengths, but we see colors and still we don't have an understanding of how these wavelengths become colors. And also the colors that you see will be different depending on the what is this other colors that are there, and depending upon the what is this you might have come across this there was a couple of years ago there was this big social media phenomenon called blue dress. Have you heard of that?
Yes, I have.
Yeah, so that is another illustration where the color that you see depends upon whether what the brain assumes to be the illuminant color, how the object is illuminated. So you remember we
Yeah.
talked. So some people who are saying they see blue dress, for them the brain is assuming illuminant is of certain wavelength, whereas for in other cases it is different from. So so one thing that I could readily think of is What is this? See, one is as they look around. Of course, this is a very difficult problem. RB did work on this problem long time ago. So when you are given a picture, how do you describe it? Say maybe in artificial intelligence, this is also they have this give a caption for the picture. Say for example, even though this is technically difficult, but there can be crude descriptions. Like for example, if I can't see, I can have wear glasses which will do image segmentation and identify objects, and then it can give a description, and that can be, if that is fed to the to the ear. Maybe let's say if he is not he or she is not hearing impaired, then I think that might help. See, the question and the problem is not at the receptor level, but at the second stage. Measurement is followed by interpretation. So this stimuli has to be, once they are measured, this many photons of this wavelength versus that many photons of that wavelength. That part can be done, and then using maybe the currently available technology for scene detections. Based on that, if that can be, if it is, if the scene is given, visual scenes are given verbal descriptions in real time, then that kind of artifact, glasses, which can recognize objects and tell them, that might help hasten or expedite the learning process that is required, that is needed for the newly endowed, newly acquired visual faculty, visual perception. So simply, if simply replacing the retina or degenerating photoreceptors have not been very didn't give like didn't give rise to immediate or recognize immediate ability to see the world as a world of objects or categories of objects. So you need that either what is this? Some translate that visual thing into other modality, and that is working. In this case, it is let's say audition. Did I answer your question?
Yes, so it was about mostly the receptor end of things. I also wanted to know about the transmission per se. Like that day we were studying about the structure of a nerve cell, right? So the way it transmits information, it has myelin sheath, which conducts the information to the next cell. What if the body, like what if something happens to the myelin sheath itself? And all the messages that we're getting from the receptors cannot be transmitted to the brain.
Yeah. So I think those disorders related to myelin sheath I'm not sure you can it has to be addressed what is this medically meaning? Rather than, I don't think it can be simply compensated any other way other than uh, uh, ameliorating the degeneration of the myelin sheath and so forth. Uh, I cannot right now remember, but there are some pathologies related to this myelin sheath degeneration. So these are all these glial cells. So for that, I don't. I think there is no way around. No way out of. It is a more of a medical problem that has to be addressed, treated by a doctor, rather than. As of now, I can't think of anything that would. Even the diagnosis of certain pathology as being related to having to do with myelin sheath degeneration, I think it would require hospital visit and. Uh,
Mhm. Okay, I get the idea.
But you have something specific in mind, like
So I know somebody who is affected by multiple sclerosis, and that means the degeneration of the myelin sheath and like eventual degeneration of the brain cells. So I was just thinking, if we see that as a disability, how can we design for people better and in the end make better products? Like, going by the theory of designing better. So that was my question actually.
multiple sclerosis. I'm blanking on the symptoms. Can do you mind telling me what exactly are the symptoms?
It starts with losing motor capabilities. So a person is not able to move their hands or legs that freely as before, depending on how the disease starts and from which area it starts in the body. And eventually it moves up the spinal cord and starts affecting the brain. So maybe it has very varied symptoms, so it depends from case to case. It's quite subjective as far as I've researched, but yeah, some people have like short-term memory issues along with the motor disability, and some people have long-term memory issues. So it remains to be seen how the brain actually gets affected and displays the symptoms, but it starts with losing control of motor senses. Yeah.
It also affects the vision.
Yeah, gradually it affects the vision.
Yeah, right now I can't think of anything. But if I think of something, I'll let you know.
Sure.
Multiple sclerosis, no?
Yes.
Mm. I think I'm not. There are a lot of this say for in a case of say motor problems with motor control and all, say I, there are all these technology brain machine interfaces which can translate thoughts into actions like but but I think most of them are very there is one Indian guy who does, I forgot his name, but he does a lot of work on this brain machine interfaces, so that even thoughts and intentions can be translated into action. Uh, uh, I think it involves wearing a electrode cap on your head, and uh, that can and that can be used to translate thoughts into actions. Like, say, even in cases like, at least in some cases, like where they cannot express in any way. And in some patients, it has been shown that one could sort of read the mind and say completely, even in case of completely comatose patients, it's possible to read. Is, simple thoughts like get me a glass of water, um, or even typing. Say maybe if in the case of motor disease, if the speech areas are spared, then of course you have the speech and this technology that transcribes speech into text now. That I think is very widely used. Even a lot of people who are a lot of people. One of my friends, he is in a math faculty in MIT, and he can't type because of maybe he has been typing too much. So now the only way he can respond to emails is by using the speech to text transcription system that is plugged into his computer. Um, But to narrow this question a little bit, in what way you want to help them? Meaning, huh? uh, uh, say you have what is this multiple sclerosis patient who is exhibiting certain symptomology, a certain set of symptoms. So, is there anything that is much more that is very debilitating and coming in the way of doing everyday things, and it is in. So, is there any specific specific aspect of that you would like to add, take help with?
Yeah, so it's mostly related to motor abilities.
Mm.
So to aid people in walking where they want, or sitting up, and basically motor abilities. Along with, in the end, when the vision also gets impaired, if something can be done in order to compensate and prevent that. Yeah, so these two things. So designing products which can help with that. So I think you kind of answered when you said there are products which can translate your thought into what you want to do, like the cap that you mentioned. So yeah.
Any more questions, comments, suggestions? Shall we start?
Yes, right. We can start with this.
Okay, so today we'll look at learning and memory, and space and time. So how these are represented, and we'll look at what is this? And then I at some point in the day, I want you all to engage in some sort of loud thinking collective meaning. So each one of you, as I was as I might have told you in the beginning, every time I teach, I also try to write an assignment of what more I learned or what. So this year, I'm still struggling with how what is the difference between experience and user experience. So designing user or user experience in general. So whereas one thing that came to my mind is many experience of a conscious experience is in some sense there is a monitor in front of me, and I see when I open my eyes and look at it, I see a monitor. So this visual experience can be thought of as a model or a representation of a thing that is in there in front of me. So experiences can be thought of as representations or models of things, and so that and then the question is, how do you go from a thing to a model of a thing that we have an understanding. There is a clear and definitive theory of going from a thing to the properties of the thing and to the conceptual concept of a thing based on which would be and the properties and then mutual determinations, and then interpreting this thing into some background or intuition to give, which would give rise to the experience or representation of the thing. So basically, now when I think of user experience, so when you think of user experience, correct me if I'm wrong, you start with an experience, if I'm not wrong. Like, in other words, you want to, let's say, what is that? I want to make people happy. So in other words, there is this experience of happiness that you start with, and then if the experience, if you think of it as going from energy to experience, meaning and the photons or whatever mechanical, chemical, or whatever energies that are transformed into the sights and sounds that populate our everyday experience. Then the question of, you say, if you start from user experience, meaning you are starting from a representation, and from that representation, you want to, basically, you are starting with experience, means you're starting with a model, and then you have to break down into basically, this is what I mean by loud thinking. First I have a model, and then I have to break it down into. I have to translate somehow and find an inverse and get back to an energy from this experience to energy, and energy meaning artifact, material thing. So basically, I have an experience which is a model. So models are usually, if your intuition or background, or background meaning the white paper in which you write, you can think of it as. So experience is the thing that is printed on the white paper. So then, what is the thing that is, so you have to separate out the white paper from the thing that is printed on the white paper, and then that is the thing you want to. That is the concept of cat, or a concept of dog, or concept of happiness, and then that you want to. Uh, uh, present it as an artifact, meaning a material thing. And when the user looks at it, it would elicit some experience. Then you would look at that, you would compare that experience with what you pre-conceptualized, or what you intended to elicit. Say, for example, if I wanted to make you happy, then somewhere, one way or other, let's say, I came up with the artifact of a picture of happy people, and I happy faces, and I display it on the, say, wall. And then as people walk by looking at those happy faces, then I can look at their faces to see if that is eliciting happiness. So, this is what I would like you all to at some level. So this would could be wrong, right, or needs refinement, needs elaboration, and there is everything that there is a lot more required. But this is what I would like each one of you to do at some point during the day today. And then, since earlier yesterday somebody started with raised the question of contradictions, and so I will give a real life scientific. In science, lot of times you come across uh, findings that might say, for example, in the present context, you might hear somebody say, "Oh, you have to wear face mask to protect yourself," and then somebody might say, "Oh, if wearing a face mask for 1 hour or 2 hour or 1 day or 2 day is fine, but if you do that all 24/7, 365 days for years on, then you'll be breathing your own carbon dioxide, and that is very dangerous." So, how do you resolve these kind of mutually contradictory? Some may not have any resolution, but some I will give you an illustration where you can have these seemingly contradictory things can be how you can see them as a unified or different sides of the same coin, so to speak. So these are the three things that we are going to do today. So first, sorry. So first, Maybrit Moser got Nobel Prize. Her husband also got Nobel Prize, and along with John O'Keefe, they worked on the place cells, meaning, and this is relevant for wayfinding and how to design spaces. Earlier, somebody was talking about what would this space that we call that feels like a home versus that feels like a resort where you go to relax, or that feels like a space where you feel like doing something productive, like working, and so on and so forth. So these guys, these three guys got Nobel Prize for this. So I thought we'll watch this lecture. This is not Nobel lecture, but a more recent one. So is it okay if we watch the video?
Sure, sir.
Yes.
I think I turned on the share sound also. So hopefully, if you can't hear or if it is not visible, let me know.
Yeah, sure, sir. We'll do that. You can hear loud and clear.
Thank you so much. This is the warmest welcome I've ever got, so I will remember this. And before I start my talk, of course, I would love to say thank you to the organizers, to Adam, who asked me to join, so the Nobel Initiative, AstraZeneca for paying, and for the university here, and for the dean organizing this, and to all of you that you want to spend 1 hour with me and try to understand what we are doing far north. So I hope you get some ideas. So I'm interested, and I hope that when I'm done here, then you have some ideas how memory is encoded in the brain, what is necessary to make memories. And I give you some cues here. Space, time is important. Once upon a time, there was a tiny girl, and she had one mission in her life, and that was to try to understand the word. And it developed into this idea, I want to understand how the brain is generating behavior, memory, our cognition, our emotions. And do you know what happened then? This girl, she was so lucky that she became a scientist. And as a scientist, she was allowed to ask these questions in the lab. And what also happened was that I experienced something that I want to share with you, because it's a way to show you one of my memories. It happened in 2014, and I was sitting in Trondheim. I was talking to the people in the lab. We had an exciting discussion, and then I got a very annoying phone call. And I understood after a while that the message was not annoying. Okay, so this was a memory I wanted to share with you. And what is important here is that this is an example of a very strong episodic memory. And Endel Tulving, in the 70s, he said, if you want to study episodic memory, there are 3 key questions that you have to understand. And if you want to understand how the brain is dealing with this, then you know a bit more about episodic memory. And that is what I hope that we can do today. So there are 3 questions. Where did it happen? When did it happen? And what happened? What we also know is that there is a brain structure just inside here, in the temporal lobe, that is called hippocampus. In fact, there are 2 of them. And it's called hippocampus, as you know, because hippocampus is seahorse, and it looks like a seahorse. So this hippocampus here is dissected out by a professor in Hungary, Laszlo Serres, just to show how similar the hippocampus is to the seahorse. What is important is that even in the 50s, we heard about this structure, because there was one man that you probably have heard about. He was called HM at that time, Henry Molaisen. He had to remove both of his hippocampi because he had epileptic seizures, and the medicine didn't help him. And then they did some experimental surgeries and to remove the hippocampus. He woke up, and he was a very nice guy. So he was social, he was laughing, he looked very normal. And then they started to test him with the IQ tests, and he was better than before because he wasn't disturbed by the seizures. But when people came back, even after 15 minutes, he said, "I've never seen you before." So he couldn't encode new episodic memory. He couldn't even know how to find the bathroom. So he didn't have the where. And he didn't have the time because he didn't know when things had happened and in what sequence. But he could learn other things. He could learn to write from a mirror, and he could learn quite some things, but everything that had to do with episodic memory, even to learn that his uncle had died, he couldn't encode into his brain. So in our lab, we started with where. But before we started with the where question, there's an important man besides my ex-husband, Edward Moser, and that was my supervisor, John O'Keefe, who we earned the Nobel Prize with. What he did was to ask, there's something special with this hippocampus structure, since it's so important for HM and other patients. If you don't have it, you can't encode episodic memory. So he had then a rat, and he had small sensors close to the cells in the hippocampus. And the rat was just walking around happy. So in his lab, they got rice. In my lab, they got chocolate, of course. So what happened is that he was just studying this rat running around chasing rice or chocolate. And then he asked himself, if I listen to one hippocampus cell, what is this hippocampal cell reacting to? Can I understand that? And I'm going to play the video, and then you can see yourself, even though I have given you a cue here. So now you hear one single cell that is active. And you see the rat is doing exactly the same thing, chasing chocolate. So what is so special about this corner? We even use this color code to show you that there is something very hot in that corner. This cell is responding to something in that corner. And then could it be that there's a special odor? Could there be some scratch in the floor? What is it? So John and others recorded many cells, and they found out that each cell had its own preference, where to be active and where to be silent. And if you record more than 100 cells at the same time for some time, then you can predict where the animal is with a 5 centimeters precision. That's quite remarkable. So just by listening to the cells of this animal, you know where the animal is with a precision of 5 centimeters. That's scary. So for Edward and me, it really started when we could have our PhD defense in 1995. So here you see Edward, and here you see me, our oldest daughter, and our supervisor, Per Andersen, and here we have John O'Keefe. So the link to go to his lab to learn this technique, to learn to record from these single cells in the hippocampus, that was quite easy. So at that time, we brought 2 children with us, and we brought this technique to Trondheim. So the first question we had when we explored this was exactly what I asked you about. Why are these cells active, and where is this information coming from? Is it so that the information is processed within the hippocampus itself, or is it fed in from a structure, brain structure, outside the hippocampus? And in order to address that question, we had this fantastic student who took his medical PhD and medical degree at the same time in our lab. And this is a rodent brain. In fact, this is a rabbit brain drawn by my supervisor, Per Andersen. You see, if you lift the cortex, then you see this beautiful structure. It looks like a sausage. But it's beautiful. And if you then slice it, you see these lines of cells here. And the information in hippocampus is coming from entorhinal cortex. It's going through this loop here in dentate, up to C3, and then up to C1, and then back again to entorhinal cortex. So we asked, is it so that if we block this loop in the hippocampus, do you still get play cells? Yes. So here are examples of 7 beautiful play cells, even though we cut this loop in the hippocampus. So what did that tell us? The information that these cells use to generate the play signal has to come from outside. And our best guess, that was entorhinal cortex. So then we took our sensors, our electrodes, and we decided to place these electrodes in the entorhinal cortex. So this is a brain, rat brain, that you see from behind. The cerebellum is removed, and this colorful area here is the entorhinal cortex. The right one, that is the medial entorhinal cortex. And at that time, we collaborated with Menno Witter, who is now in Trondheim, running his own group. And we decided to put our sensor in the most dorsal part of the medial entorhinal cortex. People had been recorded up before, but they had recorded down here and concluded that there's nothing interesting. So now you have to see yourself. I'm going to play another video. There's no sound, but there are some white dots telling. Do you see anything? Or there's some strong light here, so I don't know if you see. You see it? Okay. So you see these white dots? And this is the activity of one single cell in this area, in the entorhinal cortex. And you see that it looks not so nice. So maybe these people were right. There's nothing interesting in the dorsal, or in the entorhinal cortex. But if you have a big enough environment and you have really happy animals that love to chase chocolate so that you get enough coverage, you get a pattern that looks like this. So this is in a 150 by 150 box. These black traces, that is where the trace of the animal, and the blue dots. One blue dot, that is one electrical potential from these cells. And what you see here is that there is a very regular pattern. And this is biology. So it shouldn't be so that you can fit in equilateral triangles into a pattern that is generated from biology. And remember how deep the cell is in the brain. So we did this together with Marianne Fyn and Torkel Hafting when Marianne was a PhD student in our lab. And we decided that this cell, which is sending information to the hippocampus, might function as a 2D ruler, a metric system in the brain. What is fascinating is that when you go from dorsal to ventral, then it's like if you draw these grid cell pattern on a balloon and you blow up, then there's an expansion when you go more and more ventral in this structure. That you see here. So cell 1, very dorsal, small fields close by, and then bigger fields further away, and then you have fields that are really big and quite far away. So why do I tell you this? Because I want to bring you back to hippocampus and ask, how can you come from these grid cells to a place cell? Is that possible? Because that is our hypothesis. Yes, you can. So if you align these grid cells on top of each other, you just do a normal linear summation. Then you see here in the middle, there's a bump, but at the edges, everything is cancelled out. And what do you get then? You get one bump, as we saw in the hippocampus. I have another question to ask too, and that is, how can this cell be so precise? It doesn't have eyes, it doesn't have ears, it doesn't have a nose, except on the animal, of course. So what type of information does this cell need in order to be so precise? It needs to know the direction of the animal, and it needs to know the speed, because when the animal is eating chocolate, it's stopping, it's running fast, and it's changing position all the time. So then we asked, are there such cells close to the grid cells? And Francesca Sargolini from Rome, she came to our lab and she showed that there are some cells close to the grid cells that we call head direction cells. And they were not discovered by us because they were discovered in another brain area by Jim Rank and Tauby. But she found them in entorhinal cortex, medial entorhinal cortex. So these cells work like this. So when the animal is going west, then it's pop, 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 pop. And then there's another cell. So it doesn't help if the animal is going only to west, but it has to point north in order to get this cell to be active. So this information is then given to the grid cell. But we also need information about the speed of the animal. And then the question this Argentinian postdoc had from Patagonia, Emilio Kropf, he asked, is there a speedometer in the brain feeding the grid cells with information? And he designed a car for the rats, because if you want to find cells that respond to the speed of the animal, you have to control the speed of the animal. And then if you can decide the speed of the car, then it's fine. But it's not completely fine because the animal might fall asleep. So how can you keep the animal awake? Animal has to run. And that is why we have the Flintstone car here, to remind us that the animal is running. And in this way, Emilio could change the speed of the animal as much as he wanted. And I can show you one example of such an experiment that he did. So here the animal is running north, and here the animal is running south. These lines are just in individual laps that the animal is running, and these red dots, that is just where the animal or the cell is active. And you see when the speed is high, the cell is very, very active. Boop, 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 boop. And when the speed is slow, the cell is almost silent. And it doesn't matter whether the animal is driving north or south. The cell is signaling exactly the speed of the animal. And that is what we see also in 2D. So when the animal is just running, chasing chocolate in a box, like you have seen now so many times on these videos. So here we have 12 cells, and what you see here is that the pattern, the firing pattern is not interesting, but what is interesting is to see the correlation between the activity of the cell at the y-axis and the speed of the animal. And you see it's a beautiful linear summation. So these two types of cells, they inform the grid cell about the speed of the animal and the direction. Then we asked the question, how is this process running? And we had two Chinese postdocs, Miao and Chichen. And we asked together with them, is it so that the GABAergic interneurons, are they involved in the precision of the firing of the grid cells? So to address that question, they ordered Cree mice that had a Cree activity on the parvalbumin cells and also the somatostatin. And they used this artificial receptor technique, the DREAD technique. So you just use a virus then that will go only to those cells that has the Cree tag in these mutant mice. And then you use an artificial ligand like the CNO. And then you can activate or inhibit only those cells that you are interested in. And that is exactly what Miao and Chichen did. And this is an example of 3 cells. No, one cell, sorry. So this is before they gave the artificial drug. This is when the drug is working, and this is when the drug is leaving the body. And you see that when there is drug in the brain of this mouse, this grid cell doesn't look like a grid cell anymore. And if you ask the grid score how much grid-like is the cells, then you see that when this drug is working, then we have much fewer grid cells in these mice than before. And the same thing happened with the speed. So you see here's the speed signal. When they have the drug, it's flat. When the drug is out of the body, then there's a speed signal. And that is summed up here. And you see that the speed score is very, very bad when this drug is working. And then, so what we showed here is that the parvalbumin cells are giving inhibition to the grid cells to keep the correct position of the fields. And the somatostatin, they did not have this effect, but they worked on different type of cells. So I'm sorry that I'm rushing because I have so much to tell you, and I know that I have limited time. So you have to shout at me if you think that I should move on or go a bit slower, or we have some questions at the end. We also want to compare the different maps in the hippocampus and the entorhinal cortex. And what is that very, very different, these maps? So the grid maps, they work together, all the grid cells from the same module. And it's like a mathematical paper that you bring with you. You might rotate it, you might shift it, but it's always the same. And that is illustrated here, that you go from room to room, and it's absolutely the same map. This girl, Charlotte Alme, a Norwegian PhD student in the lab, she worked with the hippocampus, and she tested the animals in 11 rooms. And she asked, is it so that the hippocampal map is similar to the entorhinal map, that it is the same map that is used over and over again, or does the hippocampus create new maps? And what is a map in this case? It's the activity of the different cells. So what she did was to put all the recordings from the different 11 exposures to the different rooms, put them on top of each other, and then she asked, are these correlated? And you see here's familiar one, and of course the same room is very correlated. But it's a lot of blue here, meaning that it's not correlated at all. That means that cells that are active in this room are not active at all in the other rooms. Except if you go back to the same room, then even after one exposure, the hippocampus is using the same map. So to just sum it up, it seems like we have so many uncorrelated maps in the hippocampus where you can store your memory. And that is different then from the grid cells that can rather give the metric to the spatial input. So then we have to rush on to ask the next question. Are there any indication of information in the entorhinal cortex telling the hippocampus about time? Then we got this absolutely fascinating and fantastic PhD student. He came to Norway from Washington when he was 18. So he was almost the same age as my daughter. And I said, no way, this is not good. He was so excellent, you can't imagine. So he said he wanted to study the lateral entorhinal cortex, and he was allowed to do that. So that is the sister structure of medial entorhinal cortex. So the grid cells are here, the lateral entorhinal cortex, that is where he put his electrodes. This poor guy, he came to Edward and me, and he showed his results. And even though we loved this PhD student so much because he was so skilled, we said, this is not right. Because he had so unstable recordings that we said something must be wrong. And it took him 8 years and us to discuss this, and before he found out what was wrong with the data. What was wrong with the data was that time had been running in between. And this is illustrated in the experiment that he and Jürgen, another PhD student, or a postdoc in the lab, did. So that was in the same room. You can have this situation where the animal is running around in this box. You can have white walls, black walls, and you run the animal, put the animal in the box, put it back again, you clean between each, and that is exactly what happened. So this is the black box, white box, white box, and then in between you have this pot situation. What happened with the cells is that each individual cell seemed to respond differently to this situation. So if we go to this cell said, I'm measuring how much time the animal is running in the box. So you see, if it's the black box, the firing rate is low and then increasing. Low, increasing, low, increasing. So it's measuring time in, or the trial time. This cell is doing something different. It's measuring the whole session, starting with high firing rate, and at the end of the last white box, there's almost no firing at all. And another cell is telling about what color the box had, and also how much time the rat was spending in each box. And then you have the same thing here with the wall color and the different sessions. So this was the confusing part. So then Albert came up with, what if we use the general linear model to ask what can tell us best, how can explain the variance in these data in the best way? So then he asked, what about the wall color? Does that explain any variance at all? And he compared the data from the lateral entorhinal cortex with structures that we were happy about, the CA3 from the hippocampus and the medial entorhinal cortex. And not much happened when it was only wall color. Then he asked about the position. And you see that the medial entorhinal cortex with the grid cells, they signal position best of these three structures. Then he asked about wall color and position, asking, is it a white box or black box? And where is the animal in the box? Then C3 said, hey, this is my function. Then he came to lateral entorhinal cortex. And lateral entorhinal cortex was telling about time. So it was like a clock ticking, tick-tock, 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 and giving a tag to each episode that the animal experienced, each box. And this is just, when it was mixed, it didn't say much. So it seems like you have this running clock that can give you a tag to every episodic memory so that you can sort your episodic memories in the correct sequence. But as the spatial information is subjective, also time is subjective. And you know that. Maybe some of you think that this is such a slowly moving hour. And some of you think, oh, time is going so fast. And this happens also in the brain of these animals. Because when the animals were allowed to run in another task that was very repetitive, so they were asked to run in this H-shaped maze, up and down and up and down and up and down, and they got a reward, then time between each trial that they went to this apparatus was gone. They couldn't, these cells didn't tell much about that time. But now, these cells, exactly the same cells, told about how much, where in this apparatus is the animal, because the experience is telling you about time. And this experience is so different from running in these different boxes. How much time do I have now? I have 10 minutes maybe, or less? 5? 15 minutes. You are an angel. Wow, then I can relax. Ah. Okay, so then the last question. Because when we have memories, we don't only know where it happened, when it happened, did it happen before lunch or did it happen after lunch? But we also have people in our memories, we have objects, and for the rats, it's easier for us if we ask this what question with objects. So we have this situation for the rat or for the mouse, that the animal is exposed to a Duplo or Lego tower or some toys that we put in. Because you remember the other boxes, they had only chocolate. Now we have chocolate and a cue. And Jim Knierim in the States, he asked this question first. So he had these different objects. He put them in the box, did exactly what we have been doing, giving the animal chocolate. And these white circles, that is 4 of these objects. And then these are different cells that respond to these objects. And when the cell is responding to the object, you know now the code that when there is color, there is activity. So then this cell is just, pop, 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 here's an object, pop, 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 here's an object, pop, 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 here's an object. Then, how many of you remember this guy? The time guy. He is also the object guy. So what he did was again to play. So you know, it's so important when you have questions that you really wonder how to address, that you can play. So we have people in our lab playing, trying out things, and suddenly they see something that is so exciting. So what Albert did was he was recording then in lateral entorhinal cortex, and he had this situation. It was this box. He had a Duplo tower here, and he had trained the rat for 14 days. And then he said, I have found some of these cells that Jim Clearman found that respond to the object. But I want to see what happens if I remove the object. So it is like you sit in a party and you wait for your best friend, and your best friend's chair is empty. Where is my best friend? So that happened to the rat. The rat was walking here in the object. It had been exposed to this object for 14 days. And this cell said, something is wrong. Look, it's firing, even though there is no object. So we decided to call these cells trace cells, because they are the memories of what was there before. And then Albert started to play. So he moved the object to a new position. And you see that when he was moving, the trace was also exposed. So in this situation, he had moved so many objects, and this cell responded to all of these moved objects. There's nothing there. And he even cleaned the box with alcohol, with soap. There's nothing. And still these cells are saying, I have a memory for this place, there was an object. I have a memory for this place, there was an object. How much time now? 10 minutes? Thank you. Then I will end with something that is already published for some years ago, because I find it so intuitive. When you think about your episodic memory, and if you think about an odor, then it's so easy to retrieve a memory. Can you think about an odor that can give you a memory that you haven't been thinking about? Is there some paella or something that is smelling something? Oh, I'm back to Spain. So this was treated by Marcel Proust, of course, in his novel, talking about when he was a child, he was sitting with his aunt, and he got the tea, and he could dip the Madeleine cake into the tea, and it was this lovely taste. Then when he was grown up, he did the same, and he was sent back to his aunt. We studied this in the lab. How can you study this in the lab? Can you think about a task? You need an aunt, you need a Madeleine cake, you need a cup of tea. At least we have odors in the lab, and we have the rat Emma. And Emma, she's giving the task that when she is smelling chocolate, she should go to this corner, and then she's getting a reward. When she is smelling banana here, then she should go to this corner, and then she gets another reward. Do you think that Emma can do this task? It's quite complicated. Yeah? Should we see? That was chocolate. Chocolate. Banana. She's good. Chocolate. No! It doesn't help to cheat. And now she can't concentrate. So, you know, rats are like us. They give up sometimes. Banana? Yeah. And now she's back on track. And what this guy did, Kei Igarashi from Japan, was to record simultaneously from the hippocampus and the lateral entorhinal cortex when Emma and her friends solved this task. What was so exceptional with what he did was that he was able to record from the cells when Emma started to learn this, when she was completely naive until she had learned the task. So he could follow the process in the brain when these two positions were coded and the positions were associated with the odors. And this is the last complicated slide, I will promise you, and it's very complicated. But we will just go through it and then you get some idea. So this is recordings from the hippocampus, and these are recordings from the lateral internal cortex. Each line here is one cell. Here, and another cell in lateral internal cortex here. This first panels, that is when Emma is naive. She doesn't know the task at all. Or she has had some trials. And then this is when she is more skilled, and this is when she's very, very good, doing just a few errors. And this is when she's doing errors. Then you can see these colors that we have here. These colors are just the activity, the pop, 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 pop of the different cells. Some cells prefer the chocolate, red, and some cells prefer the banana. And this is where Emma is smelling the different odors. What I want you to see here is that here, the maps are quite empty. There's not so much colors. But when Emma is skilled, you see there's a lot of red activity and a lot of green activity in both structures. So that means that in Emma's brain, she has developed a map that is a combination of the odor and the place. And what happens when Emma is doing an error? Her brain is empty. Have you ever had this feeling that your brain is empty? That happens. There's almost no color, no active cells telling Emma where to go. That's life. So we think that we have now discovered cells in the brain that code for where something happens, when it happened, and what happened. And then the hippocampus is fed this information, is receiving this information, so that the hippocampus can generate our episodic memory. There are some people who can't do this. And that is those people who have a lesion, a damage, when these cells are dead. This happens in some groups of Alzheimer's disease patients. It happens in also other people. So they have problems to encode new memories because these cells are dead. They have problems with spatial navigation. They even have problems to encode the sequence of events. This is the group, how it looked like 1 year ago, only Edwards and my group. And I've told you about the people that have been involved. And of course, Edward has been involved in everything. We have got a lot of support from our local university, the Kavli Foundation, the Research Council in Norway, the European framework, the ERC grants, and Louis Chanté, Kerber, Madame Bettencourt supported us, and so on. And then I would just like to end with another memory that I would like to share with you. I'm from the coast of Norway, and just enjoy. Thank you.
Thank you, Professor Moser, for your brilliant talk, and as you can see, highly inspiring lecture. And now the session is open for questions, so we encourage especially young people, students, young researchers to ask questions to Professor Moser to use this unique opportunity. So, is there any question? There?
Can we get some light in the room so
Yes,
we see you?
now we have light.
Good.
Okay, yeah. First of all, tusen tak.
Wow.
It's obviously amazing to have you here, and I'm super excited. This might be a bit of a controversial question, but I assume most of the rats are male for like interference with hormones and so, but you were talking about Emma, and since spatial awareness is kind of like a gender issue, there's people saying, oh, you know, women have less of a spatial awareness and memory. So I was wondering if in the lab you've put in Emma and their counterpart Peter, let's say, and if there is any difference, or all brains, after all, are the same in the way that they work with these grid cells and space cells and head cells.
Thank you for asking this great question. So we had especially a postdoc from Britain, Ross Langston, and when she started in lab, she said, I'm only going to work with females because I'm going to prove that females have grid cells, border cells, head direction cells, and of course, they do.
So, Lisette Menéndez de la Brida from Cajal Institute in Madrid. Thank you for your talk. It was really clear, and I'm very glad that you can come to deliver this new data in this fantastic venue. So my question is regarding Cajal. So Cajal identified the neuronal doctrine in which the self-identity is the building block of the brain. So my question is whether do you think that the functional categories are related with the cell type specificity?
What did you say? If the functional
grid cells or speed cells?
Oh yeah, how you get functions out of the network? Is that your question?
Whether they could be related with a cell type specific of the entorhinal cortex?
Thank you for asking that question. So that was our dream when we found or discovered the grid cell, because if there would be a marker, a genetic marker or something with these grid cells saying that I'm a grid cell if you did only anatomy, that would have been fantastic. But we haven't been able to do that. So the closest we have come is this data that I showed you from the Chinese couple in the lab, where they show that it seems like the stellate cells in layer 2 of the medial entorhinal cortex are receiving inhibition from the parvalbumin cells around. And then in order for these cells to be active, they have to get information from the hippocampus. We have shown that by giving mucimol so that we inhibited the activity in the hippocampus, and entorhinal stellate cells, they were left alone only with the inhibition that surrounded them, and another input, and that is the head direction. And what happened then was that before the grid cells disappeared, some of them became head direction cells. So that is telling us something about this network, and that is also why we thought this was so interesting to see that the interneurons stained with somatostatin, they are not involved in generating the grid cells. So we are closer to this. But what we also know is that the stellate cells are also border cells, and they could have other different functions. So it doesn't even, it's not enough to just say stellate cells are grid cells. So it's still a very puzzling question, but highly exciting. So we have some interesting ideas how to solve that. And we have some wonderful, wonderful postdocs working in the lab trying to solve this question. But that is science. You never know what you get.
Sorry, I'm not young. I'm not a physician, but a physicist. So I want to ask you, what do you think would happen in outer space? I mean, the pattern, the grid pattern in a different gravitational force, in a different field, will be completely different. The man in the moon, or the man on the Mars will be different. We adapted to the new situation.
That's a wonderful question. So what happens when we go to space? Do the grid cells measure, give us the same metric? So there were some rats going out in space, but at that time they didn't know about the grid cells. They knew about the hippocampus and the place cells, but the experiments were so difficult. So it was just a short paragraph in Science telling rats have been in space. We don't know. But a related question to you is, what happens if the animal is flying? Even though it's not in outer space, but even to fly, it's a different information to you than if you count your steps. And there's a fantastic group in Israel, Nahum Ulanovsky, and he is studying bats. And when the bats are flying, he can record from the hippocampus or from the entorhinal cortex. And he has shown that the hippocampal place cell is now a sphere. And then the question is, and since you come from physics, how could you predict the grid cell, which should be a crystal? Should it be a perfect crystal? Could it be a squeezed crystal? How would these different fields be? And he's still struggling with that, because to get the coverage enough when the bat is flying, that has been difficult. But the rumor tells us that there is one 3D grid cell that looks like a crystal. But we're still waiting. Science needs a lot of people.
Thank you so much for your speech. It has been really interesting. So my question was, we don't see many female Nobel Prizes. So I would like to ask you, in your personal experience, have you ever faced any kind of discrimination against you for being a woman?
That's a very interesting question. So, you know, this little child that you saw, the little girl, she had a bigger brother, 10 years older, and I wanted to be him because he was so tough. So I never realized that I was different from the boys. I didn't think about it. But of course now, I'm a grown-up. I'm soon an old woman. And I have realized that the expectations that you have towards a man and a woman is not always equal. And of course, if you want to grow curiosity and passion, if you don't expect people to do well, then it's more difficult. But for some people, it's fuel. I'm going to show them. So what I'm saying is that in Norway, we have been blessed with not so much discrimination, and not to myself. But I am aware that we have some challenges. And what is important is that we all love science, and we need diversity. We need different cultures. We need men and women, young and old. And we need to talk to each other to solve these big questions. And I think we would do that if we work together.
Hello, thank you very much for your very nice talk. I'd like to ask, okay, we now know where the information that the hippocampus is analyzing comes from.
Entorhinal cortex.
Exactly, but where is information in the entorhinal cortex coming from?
That's a brilliant one. So, of course, we have students in our lab. One Portuguese student and one or postdoc, and a postdoc from Holland, working on, for example, the question, where is the speed information coming from? And it's not published yet, but they have been tracing the brainstem and diagonal band, and it seems like this loop is very important for the speed information. Then you have information about the visuospatial environment, so parietal cortex is extremely important. You have the postrenal important. And then, of course, the hippocampus is sending information back to the entorhinal cortex, telling the entorhinal cortex some important things. And there's odor information to the lateral entorhinal cortex. And what is so special is that, as I said, this is so far away from the direct sensory organs. So what we see in these structures is what the brain is generating from all this information that it's receiving. So that makes this even more interesting, because we go so deep in the brain and we are trying to understand how the brain is working, how it's coding, generating functions.
Hi, first of all, thank you for your talk. It was really inspiring. Some surveys have shown that younger people nowadays are less interested in the STEM fields. So my question is, what would you tell them? What would you tell them? How would you convince young people to get into STEM fields and make science?
So I think that the advice I have to both you, the young ones, but also my generation, is to give opportunities for curious people to follow their passion. So if you have passion for some questions that are extremely important for you, you can't stop it. You just have to ask this question. And that is the question I got when we got the Nobel Prize. Why don't you, do you say this, sleep on your lobe? No, can you just relax? Don't do anything, because now you're done. Tick off, Nobel Prize. Of course not, because that was not the goal. The goal was to understand. And if you have this curiosity like you had when you were a child, if you just let this curiosity grow in yourself, then you find out what is your question, what do you have passion for. And if it's not science, fine enough. But if it's science, welcome.
First of all, thank you for your talk. It was really amazing.
Thank you.
So about the other cells, we humans have a underdeveloped vomeronasal nerve, unlike inferior mammals that, of course, like the rat, has a fully developed vomeronasal cells. I'm guessing it has connection with the how do you call the entorhinal cortex? Sorry. So, I'm guessing that's the question. we should have a slightly undeveloped odor cells. I mean, like the rat. The rat could track the odor of this food, either it was a chocolate or a banana. It could track the odor and actually track it in a space. The rat was looking right at it. Us humans can't. So that's the question.
So are you asking about the vomeronasal organ, or are you asking about the experiment with Emma?
Yeah.
Okay, with Emma. So you have to remember that she got the chocolate and the banana odor in the same place. So it was this central place, and both odors were there. So in order for her to know where she should go, to right or to left, she had to make this association between the odor and the position. But what is interesting in your question is, if you did this test with me, maybe having not developed my olfactory system as well as Emma, could I do the task? I don't think so, but maybe. So, but what is important is that this is a redundant system. So this is back to your question, because into the hippocampus, you get all kinds of information. So if you, to generate, for example, the grid cell and the speed, you have the optical flow, you have the number of steps, you have also how fast you move. So if you remove the optical flow, you can still count the number of steps and you know how fast you have gone. So it is a very redundant system. So you don't, if you don't have the ability to smell something, you can still do very fine. So that is my only response.
Normally you don't, when you're in a room, normally you don't know where the real north is. So for the grid map, you need to fix a north. How do you fix it? Or
thank you for that question. It's a wonderful question. So I talked briefly about it when it came to time. That time is so subjective. And it's the same with the metric and the grid cells. So when I told you about these compass cells, I shouldn't call them compass because it's not connected to the magnetic north. So it is a map that is generated in us and in the animal. So when you come to a room for the first time, these cells just decide, this is north, this is east, and so on. And if you then go to a different room, having the same magnetic or different magnetic directions, then north suddenly can be there for this head direction cell, the same cell. So it's a system to help us to organize life. And those of you who know a tiny bit of philosophy, Kant, he said, you can't explore the world if you don't have this faculty before you start to explore. So it's just a help for us to understand the world, the mission of the tiny girl.
Hi, and thank you very much for your talk. It was incredible. I would like to know, you talk about the odors and the relationship with the memory, and I want to know if the sounds have the same effect, the music or something like that could have the same effect, could be related to the memory?
So absolutely, and even in patients with memory problems, some of their problems is to find a good retrieval cue. And then you see with really severe Alzheimer's disease, that if you play some music that they know, it seems like it can circumvent entorhinal cortex and the hippocampus that is damaged, and then go to a storage out in the cortex. So the challenge for the memory is interference, that you start to mix memories, and also to find the retrieval cue. And that is why odors are so fantastic. Because, so I was at one conference, and then John Aggleton told me about a museum in York. And that's a Viking museum, and you know Norwegians, we are Vikings. So he said, at this museum, they had the Viking exhibition, and they smelled terribly, these Vikings. And you could buy these terrible smells in the souvenir shop at the museum. So John, he went there and he bought these odors, and he met people that hadn't been to this museum for 20 years. And he exposed these people from different odors, and then he asked, what do you think about when you smell this? Oh, this is the York Museum. So, you know, odors are, even pleasant odors are very, very good retrieval cues. So you just have to find out. And that is also useful when you have to memorize things that you just have to memorize that you don't understand. And then you can use this old-fashioned technique with the memory palace, which is very useful. Then you use space as a retrieval cue. So you go from room to room and then pick up your memories when you mentally travel from room to room.
Well, we have time for only 2 questions. There was one question over there.
And then one over there.
Thank you. So I think that I understood that many of the graphics that you showed us were representing the activity of just one neuron. And I was just really curious if you could tell us how the sensors work and are showing just the response of one neuron.
Oh yeah, thank you for that question. So what we do is that we have ultra-thin electrodes. So it's a quarter of a hair. That is one. Then we twist 4 of them together, and we cut them with a golden scissor so that these 4 are so close, like this. Then we put the animal down with morphine and isoferrane, and we open a tiny bit in the skull, and then we can insert these hair-thin electrodes into the brain close to where we want to go. But then when the animal is awake and happy, running around, we can turn these electrodes down without the animal can feel anything. And then we can listen, and we can test the animal. And then finally we hear a response, pop, 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 pop. Because we have a camera above the animal when the animal is running, so we can track the behavior, and we can track the signals. And what we do is just to correlate 2 and 2 electrodes and ask how does the cell look like on this. So this is extracellular recordings. And in this way, we have these clouds of activity that are separated because there is a distance between the electrode and the different cells. And in this way, you can follow these clouds for days and weeks. And even I followed some of my cells for half a year. And that's possible because you know the signature for each cell. And you magnify the small electrical potentials 10,000 times so that you can see it on an oscilloscope and you can listen.
So we have time for one last question at the back there.
Hi, thank you for your talk. I was wondering about the encoding process. When you study those animals and you study these grid cells and place cells, about the molecular process of how do they encode. If you have performed some experiments, for example, I think about the Sina Jocelyn's papers about the entorhinal cortex and how do they increase CREB levels on those cells, and they see that they are more prone to encode this new information. If you have to think about this process could be happening at your model, because they study contextual fear memories, but this is another kind of memory, but it's very related. and also how the neurogenesis could also interfere in throughout the time, forgetting those memories that you are studying. Thank you.
Thank you for that question. So I can't answer you so much because we have a postdoc in the lab who is applying for a European Research Council grant. He's the most fantastic postdoc from Italy. And so I can't tell his proposal, but he's going to study exactly this. How do you set up these networks during development? How can you select the cells that will become place cells that are active in these different rooms? How is the brain dealing with this? So I can say that there will be answers to those questions that you have, but I can't tell you how he's doing it. I'm sorry.
Thank you very much.
Ya tienen que pasar. Nosotros ya nos vamos, ¿no?
I'm trying to get out of here. So, do you want to share your thoughts on this? What do you think of the talk? Was it useful or informative or not so much so?
So I'd like to ask this thing. I mean, in relation to humans and rats, like, is it something that has to do with a person as well to understand space and time rather than just the experiments that we saw, kind of? Because some people who are born in a certain place, like say a hilly place or something, they'll be able to navigate through hills much better than a person who hasn't gone through the place already, right? So I just try to understand whether it's also a cultural and a genetical thing.
Yeah, I'm sure. Sorry. That this is often, I think, people in a different context, of course. Both Russians and Americans learned this the hard way when they went to Afghanistan. And because they could not navigate the hilly terrain the way Afghans. I think they often say the trouble they had with when it comes to Afghanistan is the terrain that is very difficult to traverse. But Afghans having been there, it's part of their even for like say for here crossing the streets in Matigiri, like people who are used to yielding, they will be yielding forever. And sometimes it feels like all sorts of vehicles of different sizes and shapes, they'll be coming from all directions. is a more sensible way to cross the street under such conditions seems to be simply close your eyes and walk in the direction you want to go. In trying to so I think there will be a lot of cultural aspect to it. Is that your question?
Yeah, sir, I'm getting it. Because this studies do show some results, but I don't know whether that alone can be taken as a parameter to understand space-time and navigation in humans.
You see, navigation, when we talk of whether it is space and time, we are not talking about empty spaces or quote unquote empty time. They are filled with objects, be it space or time. So navigation is in a sense navigating around or through the what is this? Through the around the objects that are there. So space, in some sense, is I think she briefly mentioned in passing that one of the memory techniques that very ancient memory techniques is to imagine a big building or big an enclosure with different halls and different rooms and different number of floors, and your imagination is the limit. And then say for example, if you want to learn a lengthy huge text like and you're familiar with NID, then one memorization technique, very ancient memorization technique, is to imagine say, Rig Veda is on the ground floor, Yajurveda is on the first floor, or different Vedas are on the different floors, and different parts of the text being in different rooms. And so that is one. That is the reason one thinks of hippocampus not only as a spatial representation, but also as a cognitive map also, where things are organized. Like the experiences and so on and so forth are organized in a spatial. See, the way we can there is space and there is representation of things in space. No, we use like like curves and trajectories. Is that what you're trying to get at?
Yeah, sir, sounds good. Thank you.
I had something to add, if you don't mind my stepping in. But Santosh, there was this study on those London cab drivers. That was really interesting because it directly tied in navigation and how that works. And more importantly, like how it was done by this neuroscientist called Eleanor McGuire. And when she started, she actually got her inspiration came because she realized that there are some birds and some mammals who actually they cache food. So they put some food somewhere and they will go back to that position to dig it up later. And that's sort of interesting because even in the video we saw today, that's what they did, right? Because they would introduce these olfactory smells, like there was chocolate and there was banana, and then there were two spaces that the rat had to go, and that's how they were navigating how olfactory senses relate to your spatial senses. So that's also sort of how Elena Maguire had started, like that was a starting point. And then she started thinking, is that also something that your London taxi drivers are doing? And she has so many findings. I think she did multiple studies on London taxi drivers. And but anyway, her findings, one of them was that the question, her hypothesis was that, do they have a larger than average hippocampus, like what today is where we were discussing. And she found that there are two positions to it, and also there is this concept called ballooning of your hippocampus. So her study encompassed over 4 years, and the findings are very interesting because apparently this does exist. Your hippocampus can start ballooning after a time. So how we're talking about, like, yeah, if you grow up in, like, how ethnicity or your origin deals with it, I think it also has to do with your everyday experiences and how that's triggering it. So you could probably reference that also. It's a really interesting study. So it talks about how is there a similarity in age, education, intelligence, and does that affect it? So they had multiple findings, but yeah, I think that could also possibly be related.
Yeah, I'll look into it. Thanks, Nidhi.
Yeah, then that study got a lot of press. Cab drivers. Any more comments, questions? So, what is that? So, shall we move on?
Yes, sir, I think we can.
No. So this is, of course, this is probably you're all alert, but there are some missions I am on. One of them being to dispel this myth that mathematics has to do with quantities. So mathematics is not a study of quantities, but is about study of about qualities emerging from the variation of quantities. And so a lot of times people tend to say I'm very quantitative, I'm very mathematical, as if that is some kind of a badge of honor. But I like to take pleasure in pointing out that it is the qualities that really mathematics is maybe back in the days of Greek arithmos, arithmetic was a big deal. But now when it is all about mathematics is all about qualities. And one of the reason I bring in this in the context of studying experience is because experience is one. If you ask why anybody who's studying consciousness, why is that there hasn't been any real progress, serious advance or substantial progress? Basically, if you look at as with the case of anything, if you take reality and be analyzed it into ontology and epistemology, and ontology went on to become physics and science, but epistemology or or that knowledge, how we acquire knowledge, and of which experience is a part, still is in the realm of philosophy and hasn't matured to the point of becoming a science. And then, if you ask why is the case, why is it the case that notwithstanding all the computer simulations and AI people jumping up and down, why is that we don't have any serious advances? They will say, oh, it is qualitative, it is not like physics point. Or they will say it is all subjective. So both of these are what is this? Are lame excuses, because physics is about quantities. Even if you go get to electromagnetic field theories and fluid mechanics and all, there you come across all sorts of structures, all sorts of qualitatively different structures. And subjectivity is part of mathematics too. So that's the idea of what is this? Like, say, for example, depending on what probe you use, you say, for example, in the case of graphs, if your subjective instrument of probing a graph is a dot, then you can see dots. If your subjective instrument is a loop, then you can see loops. So if it is an arrow, you can see both arrows and loops. So the subject, that is also another lame excuse that they give. So this is my quail story for now. So I think I might have mentioned about this, no, in the first class itself. So basically, quantities are ground zero, or the most crudest account that you could give. Of course, the crudest account it turns out to be the coolest one, according to this transcription. But that is useful. I'm not denying the utility or value of the quantitative values that the property takes. Qualitative structures on which quantitative properties that can be defined on qualitative structures as a way of getting to know the. But my claim or my point is that that is just a beginning. So as you can see, even a week, going from this is a circle and this is a spiral. So here there is a lot of refinement, even everyday notions that can go beyond in some cases one needs to go beyond quantities to really get a grasp of what is going on in the world around us. Okay, so now I have since you are all interested in some sense creating experiences, I thought it would be good to have some at least baseline understanding of what is the relationship between the three things. One is the thing that is in a consciousness or conscious experience, seeing, and the other thing is that is in your mind, thinking that goes on, and the other one is feeling, or how emotion, what is the relation between these three things. So these are the, so when you speak of user experience, are you thinking in these terms, or like do you want, do they figure in your characterization of whatever desired user experience that, or whatever pre-conceptualized experience that you want to elicit? I'm guessing that seeing how given when given an artifact, I'm guessing you'll be thinking about what is that he sees, what is that he thinks, he or she thinks, or what is that he or she feels. Is it the case? You can like look at your one way to, is from everyday experience. So let's look at feeling and thinking. Uh, uh, and let's look at one specific feeling. Say, for example, you're angry, really mad as hell. Then, if you look at that, I'm guessing when you're angry, your thinking gets, your thoughts will start racing. Is it the case? Or at least uh, do you, so to make the long story short, since I guess everybody is waiting for to go for lunch, so the hint is Dalmatian and trolley problem. To see the relationship between seeing and thinking. Dalmatian is the hint, and to see the relation between thinking and feeling, trolley problem is the hint. So I give away the answer. So thinking changes the way we see, and feelings change the way we think. So if you want to change the way somebody looks at something, then you have to change the way he or she thinks. And if you want to change the way he or she thinks, you have to change the way they feel. So feeling is in some sense very important. Oftentimes, people tend to think when it comes to doing science, you have to be objective. You don't have, you shouldn't be a guy. There is no room for feeling. Feelings come in the way of reasoning clearly, or of course. So the issue is not that may be the case in the case of one particular feeling, and but thinking should always be guided by feeling, and in the sense of a feeling of respect for the object of your study is all is very important, and most serious, or at least the scientists that I know and worked with, they have a sense of, they have a feeling of respect toward their object of study. You cannot have some careless attitude or what is this disrespect for the subject of your study and expect. So in a sense, you have, if you love it, it will reveal itself to you. It will it's see the way you get to know about things. Say if you express feelings of love and affection and care towards a person, then that person might feel comfortable enough to reveal more about. The same goes with the world is in some sense like a person, whether you like. So if you want to find more about the world and how to make your way around or make things, it should be an attitude of apathy or disinterestedness is are not the things that I prescribe. In fact, there is a no, Rod McKinnon. He got Nobel Prize for crystallizing transmembrane proteins like ion channels. So he was he used to be he was a professor way back in the 90s and probably one of the youngest faculties in neurobiology departments, and he did fantastic work on potassium channels. Uh, and he used to do study potassium channels using electrophysiology. See, these channels, by virtue of being conducting ions, you can characterize the shape and size and how it is. You can pretty much see it by using different concentrations of different ions and looking at how measuring the current flow and voltage change. So he pretty much had a picture of how big the iron core is, what is the narrowest part, what is the widest part, and all. So pretty much he had a pictorial depiction, but he actually didn't see the ion channel because it has not been crystallized. So he wanted to make sure, he wanted to see it. See, he pretty much had a visual image of it based on electrophysiological recordings, but he wanted to actually see it using crystallography. So he went and talked to a couple of people, and in fact they were in Harvard at the time. There were some crystallographers who got Nobel Prize, and it's written in the textbook that you cannot crystallize transmembrane proteins because in order to crystallize you have to wash them with a detergent, and transmembrane proteins are basically oil. Transmembrane proteins are oily. So if you wash them with a detergent, the way when you wash use detergent to wash clothes, basically it breaks apart the oily substances. So that is what it does, and as a result of which you won't get the crystal structure that is known for a long time. And it has been in the textbook. So people just said, and there is no way you can crystallize transmembrane protein simply because they are oily, and in order to crystallize, you need to use detergent to wash off the wash it down, clean it up, and while cleaning it up, it breaks apart. So, but he was obsessed with that question. So he started reading, taking courses in crystallography, undergraduate courses, graduate courses, And then he was studying textbooks and crystallography. Then slowly people like postdocs and students who come to work with him, they come to his lab because he's very good electrophysiologist and a world-renowned expert on potassium channels in terms of action. So they slowly the word spread that he's no longer doing spending. So the department chair came and he said, "Get your act straight. See, you're hired because you are an expert in this. But if you want to be a novice, you can't be a professor." And basically, they fired him. Even though when you have tenure, but because of his this obsession with crystallizing potassium channels, he got fired. Luckily, one of the two guys who remember we looked earlier we talked about what is this orientation sensitive neurons that detect different orientations of lines. So one of them, Torsten Wiesel, he was at that time president of Rockefeller University. So he heard this story and he said, since he was earlier at Harvard, he knew him. So he said, okay, I mean, he also didn't believe it could be done. But since he's a very, Rod McKinnon is a very serious scientist. So he said, he gave him a blank check and at Rockefeller University. But unfortunately, even though he had as much money as he wants to do whatever he wants to do, he could not even get a technician because even technicians they go to a lab where the scientist is doing something in which he is an expert. So when he advertise for technicians or grad students or forget about grad students and postdocs because they don't want to go to a lab where the PA doesn't know what he is doing. He's still learning, studying textbook. So he he and his wife who is not a scientist, but molecular biology and especially this crystallography and all these are very laborious, manually very intensive. There is a lot of washing, cleaning, putting things, adding things, running gels, all these things. It is very manually taxing work. So his wife was helping him and within 5 years he crystallized and within a couple of years of crystallizing the structure he got Nobel Prize in a very short but when they asked him how did you do it, he simply said at every step of the process I used to ask my protein, are you okay? And that was his. So basically, he still used the detergent and all, but instead of using whatever detergent they were saying, he tried different detergents, different concentrations of detergents. So he minimized. So, but the bottom line was the point that I want to the message in the moral of the story is you have to love the subject and care about it. And so there is this myth that you have to be objective and you shouldn't let the feelings come in the way. But lot of people who made major breakthroughs, they are guided by a feeling of respect, feeling of love, a feeling of so on and so forth. I think now there is, so maybe this is, uh, does anybody want to add anything or You're more than welcome to disagree with me. Any comments, questions?
Right. I just wanted to ask, is it possible to share this content with us so we can also, go through it once again? Because some things they might require to read again and to reflect on it because,
Oh, you
the
mean the
first time.
You mean the PPTs and?
Yeah, yeah. Because
Yeah.
even the links that you shared were very, very helpful. Actually, I was able to, really relate to what, you were talking about. So it's like it allows us to do it at later at our time to be prepared or to be able to assimilate it. Otherwise, it's just words.
Yeah, yeah. Yeah. So what I will do is I'll send the PPTs now. Huh? Uh, 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 and I will also share some literature from the previous this thing. Last time when I talked, uh, uh, and there are some books that you might find relevant, some papers, more than that I shared in the abstract. So I'll do that little later now.
Okay, no problem. Thank you.
No worries. Any more suggestions? So, shall we take lunch break?
Yeah, sure.
Okay, happy lunch. See you. Will at what time shall we come back? Get back.
You can decide that. I open to.
For me, any time is okay. It's up to you.
I don't have any preference. I don't know about the others.
Two is fine.
Yeah.
Yeah.
Okay, so I'll see you at 2.
Okay, thank you.
Thank you. grabs the chair. Oh, there is one here. Yeah.
So basically, they are sensing the vibration and they are processing that vibration feedback into auditory cortex plus into the like wherever our signals go, like
So,
sensation signals go. So I was thinking about like can we I can do some research about how does it work when there is a sensory and mental stimulus, and in that case, how the other parts of the brain develop, and like how we can utilize them to do things to get them continued. So it's like let's say if there is an autistic kid and if it, so autistic kids are really good with maths, but
what was the word you said? Just
autistic.
Oh, autistic.
Yeah. So some autistic kids are really good with maths, and but they like people don't consider it as their power because they are showing other symbols of that. So can we use that one single thing so that they after a while like when they are growing up or our age, they must be wanting some respect, right? Or want to work or do something. So if their brain is developing in that area, can we develop something to help them and work around. So that was the thought basically. I don't know what I'm going to write, but this is the thought I'm coming up with, and I will do it.
So one way autistic is going to be
Not necessarily. I'm just giving one example where they're mathematically different. Some schizophrenic people they tend to remember things exactly where they are. Like let's say if I've kept the pen over here, but I'll roam around the room looking for a pen. We don't notice that, but they think about it. So it's like they have different ability where they have those tools where they find those teachers find out that, but they don't walk around it to make them independent around their abilities. So I just want to know how the brain biologically develops to cope up with their other disorders.
Most of the time, the autistic what we usually consider neurodevelopmental disorders, even deficits in hearing and they tend to be much more sensitive. Brain develops in some other respects much more than we neurotypicals. They use the word neurotypical. No? So now, so in other words, they are no longer not. Autism is no longer considered a disorder. In fact, there is one. I try to remember her name. I was really amazed. Like so, the point that she was trying to make is that you people know one language, and yet just because you speak in a different language that you don't understand, you cannot simply call us that we don't know and they're speaking nonsense. So, but the way she said that, it was like reading hand. So the way very she was very active. In fact, first I came across she was in news also. So basically, they will be throwing their hands, flailing. So that is part of their language repertoire. So, but we think if we do like this, we think they have some problem, and we put them this guy can in a box and so. But just as like if you look at Feynman's lectures, he will be flailing his hands all over the place. But since he is a MIT professor, we can't say he doesn't know what he's doing. But ordinary people, when we come on the street, with autistic or any other deficiency, so that's why the trouble with this world has been always, it is only worried about the median group, and any deviation from the medians, they are considered as not even equipped to live on the land. So I think so with respect to hearing difficulties, even though they have hearing difficulties, you are saying they sense the vibrations much more than. So one is to see that can be translated into but either area I'm trying to think what might being able to so which study we are referring to happen?
We process it with our sensory organ. So we are just taking our sensory organ. But when they people vibration, so what they studied is they applied those caps and all of that. So after that, they found out that whenever they are in some kind of vibration, they are concentrated in their auditory cortex and the sensory cortex. So they are concentrating that vibration as a sound. So when they did one study where their people candidates a rod, they asked them to place their hands on rod and asked people like us who can hear to understand what song is playing or if they can understand any kind of relevance with the songs. So they keep their hands and they just felt the vibrations or they felt like okay there is something. But when a hearing disabled person tried it, they can understand a pattern at least. They can decode a little bit of pattern out. But we can't understand it that much to that level. Because we need to
yeah. So It would be like, for example, I'm just employing. You can think of designing an artifact which can which can go which can somehow play music to this visual, auditory to those that have auditory impairment, because music is simply is vibrations. So it can be a ball or a what is this? Even something that they grab or something like this. This would then we could translate music in some sense into this vibrations of this object, and then that's since the sensory, both of them are there. So these would, I'm guessing, would translate into melody or
I'm not sure if they have to study your test, but there are lots of different methods. But the thing is, like, do we have to go on and we need to start
yeah. Yeah. So it's a writing exercise. Basically, assignment is about writing. So, but you can write about this. That's important. So you can go as far as you in terms of research, depth, and all. And then, say, for example, if the product such products are there, that, then we can think of whether whether they actually experiencing the emotions that are associated with the music. See, music, is in some sense very unique. The auditory modality, in the sense we do not have anything analogous to music in the visual modality. You probably know the story. So this painters and other visual artists, for a long time they have been envious of musicians, because you see most of the time they compared color to music and shape of an object to the meaning of the song. So in every now when we are talking, the experiences that you have or I have are mediated by the meaning of the words that we are arbitrarily encoded into that particular song. So if you record this, there will be some power spectrum in various frequencies, and that is decoded as words. And if you say, if you say, Raidu, your class is wonderful, then I'll be happy. So here, that emotion of happiness is mediated via the meaning of the words. Sound itself is not directly. So meaning is like the object in the auditory domain. So the words and the objects that you see are the auditory objects. There is a book called Auditory Scene Analysis. I forgot the author's name, it's called auditory scene analysis. So he does. Brings out the parallels between audition, sound, audition, and vision. But the point I'm trying to make is, so painters. So whenever let's say draw like this, you find a line, and if you do like this, no matter how the color, how they threw the color on the, it always conveys some shade. So it is only via the shade that we might experience some emotions. If it is a cloudy, dark shades, then we might feel a bit depressed or blue. Or if it is bright color, or if it is a seashore, but it is always after recognizing the shade. So color is always no matter how you do. So, but in the case of auditory domain, here you have sounds that have no meaning whatsoever, that evoke emotions. No, so this is so they try to, but of course they fail. That's there is a book called Color and Culture. There he talks about this. How one motivation for this abstract. So they got fed up and they started. They could not create it. They want to remove the meaning. So sort of the way a musician, when he plays some sound that doesn't have any meaning, no, but it can convey all the entire spectrum of feelings can be evoked simply by the sound. Anger, we can feel happy, we can feel sad, so all those feelings. So music in that sense is very special, and one question would be to look into it is art. Say you can imagine different musics that are known to evoke certain emotions. I'm not. I don't listen to music, so I don't know much about music except what I read read about it. But you can imagine there would be some sort of music that gives rise to happiness, some that make you sad, and some that make you reflective. And you can see if that if when they're translated into vibrations, if those that particular genre of music is translated into vibration, whether they experience corresponding emotions. That would be one thing I that I could think of right now. But I think it is in general. It is, I think it's very important that we make the world spacious enough for all kinds of people to, because there is room for everybody to flourish. So any work along those lines, I think is going to be very valuable and useful. Exactly. Your understanding, that's all I want more than anything else. Your understanding, your take on it. Not so much he said that, she said that. That is so. What we can be very critical. There is nothing wrong in being critical. I think I shared one of the articles in that about Kahneman. There I am, in fact, I sent it to science, and they said you are engaging in ad hominem attacks. Then I said, that is what he did. I didn't do that. I'm just saying what he did is wrong. Then the editor Holland thought, you both decide between you and that is between you and Kahneman. I don't want to get into this. So, but I personally feel what you take out of it, you can be as critical as you can. Because in science there is as much good as there is equally a lot of BS is going on. So I want to see yourself, your take on it, your opinion about it, your ideas about how to take it forward. That is what I want to look at, more than anything.
Also, I had one other idea that I guess the business that. So,
No, but go ahead. Oh, yeah, okay. So after the class, okay. So after the class is over, oh, so yeah, I guess everybody is here now. Huh? Or shall we wait some more time? Hello? Did you have good lunch?
Sir, I guess we can start.
So I shared these drive links. So where you'll find the literature and PPTs. So let me know if you have trouble accessing them.
Yes, sir.
You can see the screen, no?
Yes, sir, we can see the screen.
Okay, so now I'll talk about neuroscience of learning and memory. So, so one of the ways if we represent information in the context of say neural networks is as a feature vector. Meaning a vector say n elements might, each one of the elements might indicate the presence or absence of a feature such as a tail or horns or whatever might be the case. So one of the one of the computation that is being done is given some information which is given as a feature vector. The object of the neural network or the artificial intelligent machine system or whatever it is to recognize or categorize as this or that, as cat or dog or whatever might be the case. In this case, so the way it works is, as we talked earlier, so there are these feature vectors, the stimuli or input vector, which meaning say a face. The face would be represented as a vector. And elements of the vector would be feature values, length of a nose or a distance between eyes and so on forth. So your objective, the machine's neural network's objective is to recognize whether it is a face of a male face or a female face. So what it does, it will be trained prior to this testing phase on different male and female faces, basically with vectors. And based on that, it will form two categories. Each one would have a prototype. Prototype of a male face would be another feature vector. So in order to, what the computation boils down to is the test vector, the test phase is compared with these two vectors, these two prototypes, male prototype, female prototype faces. And then compared means the Hamming distance, cos theta, between these two vectors. So the Hamming distance, so whichever is closer, if the Hamming distance between the test vector and the female prototype vector is less, then it is recognized or categorized as a female face. If it is less, so Hamming distance is basically known as also city block metric. So that is what I'm trying to illustrate here. How far is Mantri Mall from Orion Mall if you were to go by the city block metric? So by city block metric, what is usually meant is that you cannot. So it is, you have to walk along the streets of the city. So for example, here to go to Orion Mall from Mantri Mall, you have to either take this road and come here, or you have to take this road and then this road. You cannot take the flight path, meaning there are some buildings or some things in between. So the distance between Mantri Mall and Orion Mall would be 2 kilometers. By going by the city block metric, and this is also known as the Hamming distance between. So this would be 0, 0, 1, 0, 1, 1, 0, 1. So the distance between Hamming distance is basically the number of vector elements that are different between any two given vectors. If you think of Mantri Mall, represent Mantri Mall as 1, 1, vector 1, 1, and this as 0, 0, the Hamming distance is, they differ in both the elements. Here it is 0 and here it is 1, here it is 0, here it is 1. So the Hamming distance is 2. That is what here I am representing as 2 kilometers. Is it clear? The Hamming distance, the idea of city block metric?
Yes, right.
Yeah. So next, what we'll do is we'll look at can you tell is Raidu more like Varahavatam or Hanuman?
I would go with Hanuman because all of us came from monkeys.
No, he never ever bring anything and everything to bear on the question other than what is in front of you. Right now we are talking about Hamming distance. That is only thing that is relevant here. Whenever I ask a question, you don't have to go any far beyond what is given in the slide or in the statement that I made prior to asking the question. So here Raidu is basically a feature vector 0, 1. And Hanuman is another feature vector 1, 1. What are the two features? One feature is bipedal. Bipedal means has two legs. That is the second element of the vector. So I am bipedal, so 1. So the first element is tail. I don't have a tail, so that is 0. Is it clear now?
Yeah. You're closer to a Hanuman because there is only one bit change.
Yeah, exactly. Yeah. So basically, having distance is counting the number of bits in which the two vectors differ. So this is the basically all that is done in all this. Basically, you'll have everything represented as a feature vector, and then you'll be comparing. So now that so objects are feature vectors, or we represent them as feature vectors. Now we look at learning. So now you have a sight, an apple, and the sound apple. So now, how does when we look at this, how do we get to recognize it as apple? Or when somebody says apple, how does this image of that might come into our mind? This is the question. This is what we are going to look at now. So remember, we were talking about neurons coding for different features, such as orientations, and some neurons coding for motion directions, and some neurons coding for colors, and some for faces. And so there are also neurons that code for objects. Like this neuron, whenever an apple is in the visual field, this neuron fires an action potential. Whenever a sound apple is made, when you hear, then this neuron B will get active. So this is the first part of the story. So there is one apple selective neuron and another apple sound selective auditory neuron and visual neuron. So now this is known as a Hebbian law, but it goes back to William James. This is from his Principles of Psychology. There he says, when two elementary brain processes have been active together or in immediate succession, one of them on recurring tends to propagate its excitement to the other. So I'll read one more time. When two elementary brain processes have been active together or in immediate succession, one of them on recurring tends to propagate its excitement into the other. So I think at this point, the notion of neuron was not at not there. People are still thinking in terms of mass action or chunk of a brain. But even in the absence of this, he did not have this conception of this came later on. But even without this conception, he was right on target. This is one of the things that you notice, like good scientists can see the future. So basically, if you translate this, when two elementary brain processes, meaning when two neurons, one selective for the sight of the apple, and one meaning one neuron which gets activated when there is an image, visual image of an apple, versus whereas another neuron which is activated when the sound apple, in response to the sound apple, have been active together or in immediate succession. So when you are teaching a kid, you might show this picture and say apple. So that is in immediate succession. It could be what he's saying, active together or in immediate succession. One of them, on recurring, tends to propagate its excitement into the other. So one of them, so what he's saying, one of them, let's say A, when it gets active, tends to propagate its activation into the other that has been activated simultaneously earlier. So now, after the child has learned, when you show this, when he sees, he or she sees an apple, he will say, "Mom or Dad, can we get the apple?" Meaning, so this sight, the visual neuron activated the auditory neuron or sound neuron, and the child could say apple. So that is how, that is what is known as Hebbian learning. This is the basic mechanism, building block of learning, associative learning. So here, later on, Donald Hebb, he's a Canadian psychologist. By this time, it was clear that. So here he says, when a cell A excites cell B and repeatedly takes part in firing it, some change takes place in one or both cells, so that A's efficiency as one of the cells firing B is increased. I see. No, one thing I want to I want to point out is, see, lot of times, this is basically a learning law that is in all these fancy AI systems. But there is always, when you say, when you come to a certain conclusion or a certain thing, there is always something, some things that are not clear to you. So here all you could say is some sort of elementary brain process. So he is not clear what exactly they are, even though he doesn't say that. But here he is explicitly saying, I don't know what change. When he says some change, I don't know what would be the nature of that change. So even when you don't have, so that is, so at any given time, our understanding is incomplete. But that incomplete understanding is also very useful to have. So at this, during the, at the time when Hebb spoke of how this learning might take place, that you see in day in and out, when you see when the traffic light turning red and everybody stopping, so you associate the meaning of stop with the color red. So basically here there is an action, a neuron, motor neuron that is controlling your action, and another neuron for color. But these two are, so he's saying he doesn't know what change that takes place. That is what I want. But then it is not like we are going to figure out everything today or before we die. So we only go as far as we can. So if we are not clear about what something, there is nothing wrong in saying, I don't know how it might happen, but you can say something, it might, if it happens, you can qualify, or under the some change, or I don't know what the change is. That is, so there is nothing wrong in, because that is the way in the science goes out. Building on brick after brick after brick after brick. So along the way, there will be sometimes it is not clear whether what how to put things together. So it is okay to say, be explicit about things that are not clear or and later on then the next person can come in. So this is so that change, what the change is basically what that change could involve, basically this is a presynaptic neuron. This is a presynaptic neuron terminal, and these are the synaptic vesicles, and these contain neurotransmitter, and if you simultaneously activate this presynaptic neuron, this neuron, color neuron, and the sound neuron, what happens is these presynaptic at this terminal, you can have enhanced transmitter release. That or alternatively, you can have more number of receptors, NMDA receptors, so that more calcium can go in, and so on. I'll come back to this little 1 second. So what is that slide should have come after this one. So what is that we need when it comes to learning that what is that red means stop. So there is some sort of temporal coincidence, or green means go. So as soon as the light turns green, everybody is moving. So the movement, initiation of the motion, cars moving, and the light turning green. So the color neuron and the motion neuron are both simultaneously active. So we need a mechanism to detect this temporal coincidence. So when you say, if you say show an apple today and you say apple tomorrow, then I don't think whoever you are trying to teach would be able to learn or associate the sound apple with the object apple. So they have to be near simultaneous or sorry. So we need a mechanism to detect temporal coincidence. So that mechanism is provided by the NMDA receptors. These NMDA receptors are on the postsynaptic neuron, meaning let's say the sound neuron. So in order for the sound neuron, these NMDA receptors to be open, they need two things. One is the neuron on which they are has to be depolarized. You remember I mentioned and neurons at the resting membrane potential would be around minus something, 40 or something. And then if positive ions go in, then it will be depolarized. And that depolarization, what it does is, under normal conditions, when the neuron is at resting state, these NMDA receptors are closed. They don't allow let in potassium, sodium, and what is this, calcium ions to come in through. What NMDA receptor, but when the neuron is depolarized, the under resting states, the magnesium ions in this neuron will block it. When it is depolarized, since the magnesium is positively charged, it will be kicked out. So in this image, you can probably see here this magnesium, the yellow one, Mg2 plus. When it is depolarized, this magnesium ion, which is under normal conditions blocking the channel, this is the NMDA receptor. This magnesium ion, basically the way it blocks magnesium ion, it's like imagine this is a freeway. NMDA receptor is a freeway, and sodium and calcium ions are like cars going by. These magnesium ions are like cows. So once a herd of cows enters the highway somewhere, then everybody, they go slowly, and the whole traffic comes to a standstill because of the slow moving. So that is what how this magnesium blocks the NMDA receptors. So once this postsynaptic neuron is depolarized, this depolarization means inside it becomes positive. Once it is positive, it repels the positive these things, magnesium from this blocking side, and in addition to this, we have neurotransmitter release like NMDA NMDA molecules released, glutamate molecules released. These are the glutamate molecules, and they bind to this NMDA receptor, and they open the NMDA receptor, and this magnesium ion gets kicked out. But since calcium and sodium, the concentration is very high outside compared to the inside, as a result of which they flow down the gradient and they further depolarize it. So basically, in order for the this NMDA receptor basically acts as a coincidence detector, meaning is by virtue of being open only when the postsynaptic neuron is depolarized, meaning active, meaning the sound apple is heard, is detected, and so this neuron got active, and the visual neuron they, also got activated, and the action potential came all the way here, and the action potential leads to and culminates in the that depolarization. Basically, action potential is again depolarization. That in turn leads to the fusion of the synaptic vesicle into this membrane. It gets fused and releases this transmitter molecules, and here these transmitter molecules then they go and bind. So basically you have both whenever you have NMDA receptor active, that means both the presynaptic neuron and the postsynaptic neuron are both active. So if you go back and look, whenever NMDA receptor is active, both neuron A and neuron B are active. So basically, we have a temporal coincidence detector, presynaptic transmitter release and postsynaptic depolarization. So now, what we have is so these are all. You remember we said feature vectors. So this is the synaptic strength. So this is the strength that increases, that changes when William James said some elementary brain process changes such that the presynaptic neuron becomes more efficacious in activating the postsynaptic neuron. Basically, it is the synaptic strength increases, meaning either the number of NMDA receptors are increased or the amount of glutamate transmitter that is being released increases. So basically, each one of this could be a neuron detecting color. This could be a neuron detecting motion. This could be a neuron detecting sound. So this neuron, what it does is it sums up each one of these postsynaptic potential, meaning when this transmitter release comes in and activates the postsynaptic neuron, there will be slight depolarization. All these depolarizations are summed together, and if they exceed certain threshold, then it goes into this regenerative state, and basically there is an action potential. So what Sherrington is claiming then, so this is the quantitatively speaking. So basically what neuron does is weighted summation, and you can take this threshold also as another input with fixed weight. So basically neuron is nothing but a weighted summation of the inputs. So what Sherrington is claiming is that all this, you remember we are talking about, we are suspended in energy. There is all sorts of energy, electromagnetic energy, that is photons, vibrational energy or pressure, chemical energy or molecules, and these are all coming through are impinging on us, on our eyes, retina, eardrum, tongue, nose. So all these are qualitatively different. So now, in some sense, neuron is a model system of the brain itself. So brain is basically integrating all these things that are coming through the ears, through the nose, through the your thoughts in your mind and your feelings in your all these things. They're combining it. Brain is putting together into the cohesive experience that you are having. One characteristic feature of your experience is that it has this it is cohesive, in the sense there are no empty spaces in your experience. If you think of your experience as a paper on which there is something printed, there is no empty spaces. Unlike here, there is empty space in which you can write something. But your everyday experience, if you think of it as a planar image, there is no empty space in your experience, and it is one unified thing. So, but they all come in as distinct pieces. Sight is different from electromagnetic energy, is different from chemical energy. Molecules versus photons versus vibrations, they are all different, but they are all put together in some form to give rise this unitary conscious experience that is your everyday experience, moment to moment experience, or waking experience, or so. What Sherrington is claiming there is saying that what brain is doing is reflected in neuron, or neuron is recapitulating in an elementary form the integrative action of the brain. So that is the reason people usually talk about neuroscience and rarely talk about brain science, except for the except for MIT. Even I think somebody has to tell them that you don't have a brain science. So the reason most people speak neuroscience and not brain science is because we haven't been able to figure out how the brain does what neuron is doing with respect to quantities. So these excitatory potentials are simply going from minus 40 millivolts to minus 30 millivolts or minus 20 millivolts, or in the case of action potential, it goes to plus 70 millivolts. There will be, and it again comes back. So we know the scientific, to the extent we have science, we have scientific account only at the level of neurons and networks of neurons. So in order to have what we are moving, what we want and what we are hoping, or what Sherrington is trying to make show, pointing out, is that a neuron can be looked up as a model system. And now the question becomes, okay, now we know how to add and multiply numbers or quantities. Now, in order to understand brain, what we have to do is I try to figure out how to multiply and add, so to speak, qualities. And now that we have a definition of quality, it is possible. And if anybody is interested in, let me know. I'll share with you a manuscript I wrote. Professor F. William Laurie, he was the one who defined the gave the axioms of. So quality is a special case of unity or cohesion. Quality is a special case, and then if you can say, for example, color is an intensive quality, meaning if you have an object, you can speak of the color at every point, whereas shape, this circular shape, is an extensive quality, meaning you can't point here and ask what is the shape. You have to take into account the entire object in order to come to in order to see the shape. Whereas color is intensive. It can vary from point to point. Whereas shape is a much more global thing, and this is a local thing, if that analogy is held. So this is another reason why we use analogies, and in fact science for the most part goes on building on these analogies. So but unfortunately, so now I don't want to go vent about why there is no progress in neuroscience, but we'll look at what little progress we have. So this is the sight neuron and this is the sound neuron, and so there is a weight. You remove this weight that is attached to the signal. So when a signal comes in, it is weighted by the signal, and then it affects or this neuron. So basically, this weight is, if you take it, this is what the coincidence detection. Basically, if both presynaptic neuron and postsynaptic neuron are active, then this weight increases. That is what William James saying. Some process changes such that the efficacy of the this neuron to activate this neuron increases. So whenever both of them are active, this is 1. So whereas if this is not active or either one of this is not active, and if you take a not active as 0, then this would be 0, and the weight attack would be 0, and this won't be effective in activating this one. So this is the basic rule. Coincidence detection or associative learning or Hebbian learning, and the weight increases in proportion to the correlated activity. So if you have that mechanism, then you get content addressable memories. Basically, this is your stimuli, 1 by n vector. In this F1, F2, Fn are basically the feature values. So for example, if it is the feature dimension of tail, I don't have a tail, so this will be 0. If it is if this is horns, I don't have horns on my head, so this would be 0. This is bipedal, then I have 2 legs, so this would be 1. So then how do you memorize this? So you remember memorization is basically forming this weight matrix. Synaptic weight matrix. So the memory is this weight matrix. So the way you go form this weight matrix is, if you want to memorize the stimuli, take its transpose and multiply it. So in other words, and recollection would be then basically if you want to say once the child has learned what is this the sight of learn to associate the sight of an apple with the sound apple, then you can present the stimulus itself again. Say if it is apple is coded as a vector, once it is learned, then you can present the vector to the system. Now this is your brain. The memory is your brain, or memory in your brain. So now you multiply this stimulus, then what you'd expect, if it is stored, then you would expect the output to be the same, recognized as it as an apple. So this is F in W is F transpose F, and now if you multiply F with F transpose, this is a number. So basically you get a scale. Since these are vectors, you get a vector in the same direction. That is that is what is considered as recognition. So basically, what happens when you do this learning, associative learning, meaning core pre and post synaptic activation, is that you form an energy surface, and these energy minima corresponds to the memories that are formed. So this memory would correspond to this energy surface. So if your stimuli is like this, is this ball in this high dimensional feature vector space, is a point. So if you put if you place show the exact image that has been memorized, that is like placing the point here, then that point will that ball will stay there only as the network iterates. But if it is say if it is slightly smudged or smeared or some noise added, that would be a little bit away from the energy minima or the memory that you form. So by virtue of the network dynamics, this learning loss, the network will be always evolving. Basically, evolving means this operation of multiplication. So what this does is, this if you start with this, after learning this or memorizing this apple image, if you smudge it or smear it, and that would be this point. This point, as a result of the network dynamics, comes down to the energy minima. So these are all known as gradient descent learning loss. Just as water falls down the gradient, or goes, or any object goes to from high energy state to low energy state, this network also evolves and goes to the low energy states. So the strategy is to make the desirable whatever you want the system to do, to make those states energy minima of the system, and such that this energy minima could be all the moves, winning moves in a chess game or a Go game, and then the system would, if wherever it starts, it will come to this. Basically, this is all that there is to it. But given the computational power that we have now, and given the cheap computation, you can have feature vectors that go 1,000 elements or 10,000. It doesn't really matter now. And you can have here, this is only one layer. You can have a 1,000-layer network. So basically, in the space of all possibilities, you can extract the part of the space. In this particular case, these two are the points that are the energy minimum, meaning the two things that you memorized. But you can imagine a situation where you can have not just points, but the surface or any higher dimensional object. That could be in the case of like Go and all. It would be some sort of higher dimensional part of the space of all possible moves that you can make. So the objective is to extract that solution space. And there are now what I want you to illustrate is so before I move on, do you have any questions or comments or suggestions, or is it clear in the first place what I am talking about? I take it that not a single word I said made any sense, huh?
No, sir. I think it's a lot of information, and we are trying to process it. So it's sinking in.
Okay, we'll come back to it, or you can write to me. I'm more than happy to elaborate on it.
Sure,
but there is something that I want to. Yesterday somebody asked about contradiction. So I want to give an illustration of how I went about resolving one contradiction or one seemingly contradictory finding. So this is related to learning and memory also. So NMDA receptors, they are involved in learning and in learning in our memory. The way you model in dish is say for example, you can take neurons from the brain of a mice or a rat, and you can grow them in a dish, and you can record from them. So if you, for example, or you can even in the a live animal, you can insert an electrode, and the way she was talking about in the early Maybrick Moser was talking about, you can implant an electrode in the brain and in the hippocampus, and you can induce LTP, long-term potentiation, meaning you can increase the weight between two neurons. So here the increased weight is measured in terms of the EPSP slope. And EPSP is excitatory postsynaptic potential. Meaning when the neurotransmitter is released by the presynaptic neuron, if lot of it is released, then a lot of receptors will open and a lot of ions will flow in, and the rate at which the postsynaptic neuron depolarizes will be faster. So it is like RC circuit. So the slope would be slope would be higher. So the way you measure LTP is in terms of the slope. So here what they did is you can do what is you can with the genetic techniques you can take this is a wild type and this is a mutant rat which in which one of the NMDA receptor subunits has been removed. So one thing that you see is there is very little learning induced, meaning the slope didn't increase much compared to the wild type. And that means there was less learning. The weight did not increase as much when both post and presynaptic neurons were stimulated. And there is a behavioral assay also. So what they do is they have sorry. They have you remember she showed a tub or a container. So you can also have that container filled with some opaque liquid like milk. And beneath the surface of the milk, in one of the 4 quadrants, you place a what is this platform on which it can sit, and that platform would be high enough that it can breathe. Otherwise, swimming takes a lot of energy. No, it can't. Rats are not like fish. They're land animals. No, there is only so much they can swim. So once it finds this platform, from next trail onwards, it will go no matter where you live. It will go straight onto the platform and sit on the platform because by virtue of the fact that it is a terrestrial animal as opposed to animal like fish or that can swim. So once again, what you see here is the latency. How many seconds? How many as the trails went on. So here you can see in the wild type, the amount of time it took for the rat to climb onto this platform decreased. So by the time it got to 12 trials, within 20 seconds, it went, no matter where you leave the rat, it climbed onto the platform. Whereas the mutant one that doesn't have one of the NMD receptor subunits, it was not that successful in remembering where the platform was. That you can see in this pathway. So here, this is the where the rat was placed, and immediately it went over try. So more you can see most of the time it is spending on the quadrant where the platform was, whereas this one it doesn't remember that the platform is in this quadrant, so it is roaming all around all the time. So this is the way we go about testing at the cellular level and at the behavioral level, whether an subject has learned or ability to learn. So what happens? There is this another thing that happens. So as we grow older, long-term potentiation decreases, meaning the synaptic strength between pre and post-synaptic neurons. And if you look at, say, this is the age of the rat. So if you look at the synaptic current, when the NMDA receptors, when glutamate neurotransmitter is released, the NMDA receptor opens and the current goes in at a certain time constant. What you notice is that the time constant of returning the NMDA receptors closing, current reaching back to the zero level, baseline level, the time constant becomes faster, meaning less amount of. This current is the current carried by the charge carried by the calcium ions and sodium ions. So the amount of calcium that goes into the postsynaptic cell decreases with age, and as a result of which, since learning involves LTP, meaning this increased. Yeah. Is there a question?
No, right. I guess somebody has unmuted it.
Okay. So basically, with age, your ability to learn decreases. So that is one find. So as you increase, as you go, as you age, the synaptic current duration decreases, and which in turn results in the decrease in LTP, and which in turn decreases in the ability to learn. So that is one thing that we'll take note of. And the next point is, you can also, this NMDA receptor has 4 subunits, NR2A, 2B, 2C, and there is another. So you can overexpress these subunits also. When you overexpress this NR2B subunit, so you see, when you think of a NMDA receptor, think of it as a beer barrel. Like if you go to any brewery, you might have. So it is like a container, like a pipe, which has a round. What is this? Wooden things, which form a circular hole, like a barrel. So you can overexpress this NR2B such that each NMDA receptor, instead of having 1 or 2 NR2B subunits, it could have 5 NR2 subunits on that. Once you do that, what happens is EPSC duration increases, meaning this one increases. So that means, as a result of which, more calcium goes in, and as a result of which there will be more LTP, meaning the synaptic strength increases, as a result of which there will be more learning. So now increasing EPSC duration increases learning, and if you go back, decreasing EPSC duration decreases learning. And now we'll look at another name. So now this is basically, I was referring, there is a CA threshold model of LTP, meaning when NMDA receptors open, the CA potassium ions come in, and the more potassium ions come in, the more LTP we have. And the longer the EPSC duration, meaning this one, the long, this is longer than this one, EPSC duration, measured in terms of time constants of returning to the baseline. So that EPSC duration, if it is longer, then more calcium ions go in, and there will be more LTP, long-term potentiation and learning. This is the threshold model. And now, now if you go and look at a NR2A knockout mice, what they have is, so you can overexpress any one of the proteins, or also you can knock out one of the proteins. But these are all proteins. So subunits of ion channels are transmembrane proteins. So if you know NMDA receptor has different subunits, if you remove this subunit, meaning you can make mice that don't have this subunit. So NR2 mice that don't have NR2A subunit have longer duration EPSCs compared to the wild type. And when you look at the wild type, now wild type has more learning compared to the NR2A knockout mice. So this is little bit counterintuitive, little bit is in contradiction, is inconsistent with the earlier version, where we here. So here we are saying as NMDA EPSC duration increases, LTP has to increase. That fits in with this one. When you overexpress NR2B, LTP EPSC duration increases, and LTP also increases. So here, when you delete NR2B, NR2A subunit, then also you are getting increased EPSC duration. That means more calcium is going in. Then you would expect more LTP, but you are finding less LTP. So these two things are mutually contradictory. Thank you very much. So now there is another pharmacological data. So we looked at developmental data, we looked at genetic manipulations. Now we look at pharmacological data. So there is this drug called memantine, which is prescribed for dementia. And what this does is, this improves learning and memory. But when you look at the NMDA EPSC current in the presence of memantine molecule, this is what I did. You find that memantine decreases the EPSC duration. So that means the amount of current or amount of calcium that is going in decreases. So you would expect in the case of memantine, the memory or LTP to be decreased because less calcium is going. But what you find is, compared to the control, memantine is increasing learning and memory. Memantine is improving memory, but it is decreasing EPSC duration. So we have two sets of contradictory findings. On one hand, with aging, EPSC duration is decreasing, and along with it, learning is decreasing. And when we overexpress NR2A, 2B, EPSC duration is increasing and learning is also increasing. But on the other hand, when you delete, knock out NR2A, EPSC duration is increasing, but learning is decreasing. And here, when you decrease the EPSC duration, learning is increasing. So these are inconsistent. These are inconsistent. So developmental and NR2B data say that as you increase the NMDA EPSU duration, more calcium goes in and LTP increases. But pharmacological and NR2A deletion data says that as you increase the EPSU duration, LTP is decreasing because in both cases, remember with memantine, the EPSU duration is smaller, and memantine has improved learning and memory, meaning the strength and potentiated or increase the synaptic strength. And in the case of when you remove NR2A subunit, the EPSU duration became longer, but LTP decreased. So these two are going in two opposite directions. So how do you resolve this contradiction or inconsistency? So forget about all the terminology that I threw at you and just look at this. First, do you see this as mutually contradictory findings? One is directly proportional. If you think as y, this is y. Y is proportional to x. Here, it is inversely related to x. So that part is clear now. Forget about everything else. Just look at this. This has positive slope and this has negative slope.
Yeah, that's clear, right?
So, so if you say, as you, what is this? If you say, if I say, the more money you have, the happier you are. That is this part. And this one says, the more money you have, the less happier you are. That's what this is saying. This is saying happiness is proportional to money, and this is saying happiness is inversely proportional to your wealth. So do you first see that they are mutually contradictory? Positive slope is mutual, is not same as negative slope. Are we on the same page, at least as far as positive and negative slopes are concerned?
Yeah.
Huh?
Yeah.
So, say for example, what is what is the current is proportional to the voltage applied. That is one statement, and the current is inversely proportional to the voltage applied. That is a opposite statement. No.
We understood the contradiction in both. We understood the contradiction in both the results. One is saying directly proportional, another one is saying inversely proportional for the same set of variables.
Yeah. So now my question is, how do you go about resolving this inconsistency? Resolving this inconsistency involves putting these two data sets together.
Yeah, and what is that developmental and pharmacological denote?
No, forget about all of that.
Okay.
There is one line with positive slope, there is another line with negative slope. So the resolution of this would cannot ignore either the negative slope or the positive slope. Okay. So it should involve both of them. It should include both of them. So what is the simplest way to put them together? So your conclusion would include both of these lines.
There is just a linear relationship. That's all we can say about it. It could be either positive or negative.
Yeah, so you are almost there. It could be positive or negative. So it could be positive at some range of EPSC durations, and it could be negative in some range of EPSC durations.
Yeah.
Huh?
Yeah.
So now, what would be the overall shape of the dependence on of LTP on NMDA EPSC duration?
Do they negate and cancel each other out?
No. See what you said. It could be either positive or negative. That is absolutely you are right on. You nailed it. So it could be. It cannot be both positive and negative at the same point. So it has to be positive at some point, at some points or some point, and it has to be negative at some other points. So some part of the x-axis it could be positive, and some part of the x-axis it could be negative. So now, what would be the shape if you combine these two parts?
It will be a triangle.
Huh?
It will be a triangle if it is like, if the first one is occurring before.
Yeah, so it would be a inverted V or inverted U.
Yeah.
So this is how it looks like. This is an actual calculation based on the data sets, but it would have an inverted U shape. Basically, in the case of, even in the case of money, if this is money, say if you don't have any money, you are really miserable. And if you are a millionaire, you are probably very happy. If you are a trillionaire, you are really not that happy, because you are more worried about losing that money rather than enjoying the luxuries that money can buy. So most biological systems have physical variables can go up to infinity, but biological systems there is no infinity. So x-axis can go on to infinity, but y-axis in biological systems, then there are two possibilities. One is either you get sigmoid shape, that is saturation, like an S curve, is slowly increasing and then linearly increasing, and it will either it will saturate or it will come back. So that is basically what is happening in this case. So this is basically a model of how you might say, for example, if you model the EPSC durations as pulses. So basically, what we are asking is, we want basically a coincidence. No, coincidence is the currency of learning. So you want to find an formula for coincidence of say there are say however many feature vectors are there, however many elements there are in your feature. Corresponding to each one, there is this one opening and closing of the NMDA receptors. So now, if you have, if you fix, say, so EPSC duration and the number of EPSCs. A temporal coincidence as a function of EPSC duration. So as you increase, So if you imagine increasing the EPSC duration to infinity, so at some, no matter once it opens, if it doesn't close, so every feature vector, once it went into the state of 1, it stays there for eternity. Then at some point you'll have all of them coinciding. The number of feature vectors that are coinciding increases. So that is what this slide says, the number of EP. So in other words, if you remember, what is this? What is? Oh, one way to look at it is like, say you toss 10 coins, or you toss a coin 10 times. So what is the probability of getting say 2 heads out of the 10 this thing? So let's say I want you take a coin and toss 10 times, and you want let's say 8 heads. So as you increase the individual probability, if it is p equal to 5, then the probability of getting 8 heads, if the probability of head or tail is 0.5, and the probability of getting 8 heads would be something. As you increase the probability of getting head from 0.5 to 0.6 or 0.7, then slowly in the probability of getting 8 heads, you reach a peak. And if you increase further the probability of getting a head in an individual toss to 1, let's say. Every toss, both sides you made head. So the probability of getting 8 heads would be 0. So it will come back down. So so basically this is nothing but an expression of a binomial theorem. So as it will increase. There is the probability of individual heads, individual events, and then there is the probability of finite number of heads. So if out of n features, you want k features to be simultaneously active, so that those features can be collected together into an object. The way we put together the sight and sound together. So in that case, we said only 2 features out of some n number of features should be simultaneously open. So so basically, I think if anybody is interested, I can send you this formula. I'm not very good at statistics and probabilities, so I could go only as far as binomial theorem. And if you differentiate it twice, you get a what is this? This inverted U shape with the binomial theorem itself. But to actually calculate this using the actual numbers, Professor Ram Subramanian here at Indian Statistical Institute, he helped me. He did the calculations and gave me the formula for this. I can share if anybody is interested. But and in fact, this turns out to be the optimal EPSC duration, and corresponds to the measured EPSC durations. I think that's all. Yeah. The reason I wanted to bring this up is this is a common theme that because most of the time you don't look at the entire range of variation. You only look at a part of the variable or segment of the variation and look. It is commonplace to find mutually contradictory findings, and those tend to be. It's not just here. In fact, at the very foundations of mathematics, there is a contradiction. I think, did I tell you this? Cantor defined element as distinct and indistinguishable. And a lot of people thought, oh, that is a contradiction. That is, we can't have contradictions in mathematics. And then they messed up the set theory, von Neumann and Germelo and other guys. So then Professor F. William Laurie came and finally he showed that Cantor's definition of distinct and indistinguishable corresponds to the abstraction process of abstracting given any category of objects. You remember we are talking about you throw away all the variation and then you throw quantitative variation and then you'll arrive at the quality of catness and then you throw away the catness, then you end up with the all ones points. Elements of a set. So elements of a set, they don't have any property by which you can tell apart one element from another element. They are all just ones, all ones. So that was thought to be a contradiction. So there are distinct, of course, on a superficial reading, distinct, indistinct. These two are obviously contradictions, but the what is this? We do come across contradictions all the time, and we need not get. But of course, we have to. We cannot simply say it is true and it is false. We cannot simply say it has positive slope and it has negative slope. But when we say it is inverted U, then it is we accounted for, we combine the seemingly contradictory things into a cohesive, sensible account. That's what the objective is. So we do not accept contradictions in the sense, we try to resolve them. And that resolution of the contradictions involves unification. So that's the unity and unity of and identity of. This goes back to Marxist philosophy also, Hegelian philosophy, unity and identity of opposites. So with that I will end today for now. Let me know if you have any questions. Comments, suggestions. Yeah.
Hi to Vikas here. So I just wanted to ask, does EPSC duration has something to do with the self-discounting aspect of reaction? Like, will the neuron be trained without self-discounting or with self-discounting? Hello?
Yeah.
Am I audible?
Yeah, I heard you, but I'm having trouble relating these two concepts. Huh? You must be very creative to connect this
I'm trying to
self-discounting
connect
to it.
everything, like what you have taught us.
So, so EPSC duration. Uh, uh, yeah. See, the way self-discounting works is basically you discount the constancies.
Right.
Whatever is constant, that is being discounted. So illuminant is always constant. So right now, the photons that are coming from that is same. So, but the photons that are reflected of the objects, that is varying. So, self-discounting means you subtract out the baseline.
Right.
So basically, imagine if I'm holding a cane all the time. So the sensation of the cane in my palm is the baseline. It never changes. So whatever changes when it hits the grass versus when it hits the tip, those would be small changes with respect to the grip you have on the disc. But over time, those small changes by way of like you can use standard nearest neighbor interactions to discount the constant. So basically, self-discounting is taking whatever is constant, whatever is not changing, and throwing that away and amplifying the small changes.
Okay. Got it. So EPSC duration is always variable, postsynaptic reaction of neurons. So is there any way that it could be constant or stagnant at particular time, point of time?
So EPSC duration. So EPSC duration is what is this? So it varies as we have seen in this all this pharmacological and all these things. So it varies, but what I'm trying to see is try to whether this whether what if any role does the variation in the EPSC duration play in discounting the illuminant or self-discounting yeah, I think right now I don't know the answer to that question. Okay. But
Maybe you can refer a book or something or material or something.
You see, there is in the folders that I shared, there is principles of neuroscience, and there is another aspect to these receptors, which is desensitization.
Oh.
So if you have, even if you have NMDA receptor not in the case of NMDA, but you can say other acetylcholine receptor, even if you have constantly putting acetylcholine chemical, receptors desensitize, meaning they will open and they will stay in the open state and they go back. Like this one. That's what I was trying. So even if you have NMDA constantly, they won't be open constantly. They will have some open lifetime average. This is all stochastic.
And then represents the desensitization of the trough in this wave represents the desensitization, right, of neurons.
If you imagine this as opening, this is opening. Right. And this is the baseline. Baseline is closed. Right. This is off, and this is on. But at the receptor level, even if the transmitter is there all the time, the receptor has a characteristic time constant that determines the EPSC duration. So this is individual NMDA receptors. EPSC duration is the summation of all these. So if you this one, I'm recording from a neuron. So the neuron has thousands of NMDA receptors, and each one of those NMDA receptors opens and closes like this. So that global EPSC duration is a function of this stochastic openings.
Okay, got it. Yes.
So, in fact, these are the ones that change. Of course, these won't be constant, but these are on the order of milliseconds, 10 milliseconds. Some of them are even very hard, below the resolution also. So, but and desensitization also at the level of individual receptors looks like the same. It will open for a short duration, like few milliseconds, and then it will close. And these two are random. And but in the absence of in the presence of this, the system is more likely to be in the open state rather than in the closed state. But that differs as a function of what is this NMDA receptor chemical concentration, neurotransmitter concentration, and so on and so forth. But what yeah, I think
Can you please speak? Huh? Did you ask me anything?
No. I'm trying. In the when you first asked, I couldn't see how you got from there to here, but now I can see a little bit. But still, I don't have the answer for your question. There, because beginning with glasses that you don't feel unless you attend to them with discounting the illuminant is one of the core computation in vision. That is the first thing that you do as part of detecting any visual object, discounting the illuminate. So, and again, that goes back to the idea of contrast being the basic building block of our experience. And so, for now,
self-discounting aspect is restricted to static elements, I guess, according to your lecture.
What is that?
I'm saying like the self-discounting aspects are only restricted to static elements, so which doesn't change, I guess.
No. You see, if you take the case of what is that? Hairdresser cutting your hair. This the scissors handle will be moving now. Across that, but
different angles.
But the nature the nature of the variation is constant.
Okay.
You are right in so there is constancy, but the constancy is the nature of the variation. Say for example, so here there is a change. This is a dynamical system. There is a change, but this change is of a particular nature, and that change is different from this change. So you can have changes, but the nature of the change could be constant.
Right.
As in the case of the movements across the fingers, that iron thing, iron, I don't know what the name of that part, that the rings, the motion, that the way they move around, that is constant.
Okay, now I got it. Yes.
So that is the idea, that you can have qualitatively different types of changes.
I think like if you relate to that to the NMDA receptors, so the millions of NMDA receptors pulses combined together and can form a constant variation which is static itself, maybe, or the change is the change of the each NMDA receptor combinedly can be also a constant, I guess. Is it like that, or is it completely variable?
No. You should remember, in biology, things can't. They're all, every changes are all constrained.
Okay.
In fact, let me see if I can find that this thing. One second. I'll illustrate. This video will illustrate very well. One second. I'll try to find this.
My job is to make college easier because students have a lot on their own.
Can you see? Can you see
the ad is playing now? The advertisement was playing. Yeah,
no.
Yeah, yeah, I can see some dots like a human pattern walking.
So Now, do you see a human?
No.
So it's only when it is moving. This also goes to the Gestalt school.
Okay.
So it is this is the idea of trying to reconstruct. You can even tell the gender of the person moving. Can you tell?
He's male.
Yeah, it is. So basically what he did is he took a person and put point lights on his what is this on various parts of his body, and so basically this is the essence, the entire this video. I'll share it with you. Oh no, go away. Where is I shared that video. I
Yeah, got it.
So basically, it is this. What is this? This is the. So both the unity or cohesion of the object.
My job is to make college easier because students have a lot on their
Sorry, coach.
plates. Like hard.
Are you showing any video, Roy? Are you showing any video to us?
I think I somehow it got paused. I don't know what happened. I think I closed it. So wait, what is that? So resume share. Now it came. Yeah. So this is basically my current research interest. So the idea is that both the way things change is limited. Things can vary. Is limited by their nature. So whether it is and by looking at how things change, you can tell the nature of the thing, just as you could readily see once once the point light walker is walking, you can readily recognize it as a male. But when you look at the individual frames, you won't be able to tell that it is a what is that? So that is again related to the idea that contrast is the basic building block. It is the change. So constancy is a constancy. Is useful, is needed as a background to build the variation. In other words, constancy is a limiting case, like the blank paper. Constancy is like the blank paper on which we paint the variation, so to speak.
Like a baseline. like a baseline.
Yeah. Okay, absolutely. Any more questions, comments? So, would each one of you go in alphabetical order or in inverse alphabetical order and say a word or two about what you are thinking about writing? Those of you who already talked can talk more. Those of you who never talk, you see, I can completely, I can wholly sympathize with those of you who are not saying a word, because I stopped going to college long after 8th class. I thought it's a waste of school, is a waste of time. So I'm really very pleased and pleasantly surprised that you find whatever I'm saying that you're listening to whatever you're saying. My mode of learning is only book learning. It is not visual or YouTube, watching YouTubes or anything. So it's okay if you don't feel like saying anything, but I'm okay with listening. There is no such thing as half-baked idea or nonsense or anything. Everything can be made sense of when looked at carefully. Because the nature of reality is such that, see, reality in some sense is a sensible thing to the extent it enters our experience. So we can try to make sense of your ideas or your whatever thoughts that might come to your mind. Say the moment I heard you speak of self-discounting and EPSC, I thought, what the hell is he talking about? But then a couple of minutes of sober reflection made me realize that yeah, it is there is something worth looking at. So please feel free to. Or you can just type in the chat, and then I will read out loudly, and that would start the conversation going. You can just type a word or a phrase or a sentence or whatever that comes to your mind. This transcription is surprisingly accurate.
What I do, what I say.
The transcription is surprisingly. I'm pleasantly surprised that it is very good.
This is an updated version of Zoom, so the transcript switches on. Right? So today, like there was a great learning that in every variation there is a constant. I mean, every variable is backed up by a constant. Without constant, there is no variable.
Yeah. So, or to put it another way, we need constancy to make sense of variation.
Also, to experience and read the variation, we need constancy.
Okay. See, we are suspended in variation. Even as I said, the molecules that constituted my body are changing from moment to moment. There is nothing. But without the constancy, basically Satir is a constancy. And that constancy is for somehow provides all the structure, that you need, or all that you need to create, to model or to represent all sorts of variations, all sorts of cohesions, all sorts of qualities, everything. So that in itself is rather, in and of itself is a very interesting notion. How, in a sense, but technically speaking, it is not absolutely constant, in the sense of, like, for example, if you so that is still an open problem. So there is still some element of very almost, you can say, zero variation, but not zero variation. In the case of sets, there, just as in the case of white paper, it is not completely devoid of color. It has some color. That is all colors combined. So if you combine all the colors, going back to the Newton took the white light and what is this?
I think I can cognitively relate this to the. Hello?
Yeah. I'm listening.
Yeah, I'm saying, like, I can cognitively relate this to the solar system, considering the sun as the center of the solar system, but it's not the absolute center, right? It can be variable based on the conditions the planet has, so the magnetism, etc. So similarly, I think the creation, the dynamics of the entire creation is also reflecting somewhere in the creation of our brains, neurons, and the system of the human being. I'm thinking.
Yeah.
Am I thinking larger?
No, I'm not. I'm laughing because you hit at something very profound. So there is this electromagnetic field that goes around, travels around the Earth.
Right.
The global electromagnetic field. And or there is an electromagnetic field in our brain that is synced with the Earth's electromagnetic field.
Yeah, correct.
And if you want to get, if you allow me to spook you a little bit more. So, one of the reasons this is for the most part of the time, most part of my life, I never worried about rebirth or what happens after death. I said I am done with death. But then,
That's it, like.
huh?
No. Continue, right? I'm listening.
So my friend Rohan, he told me that. So see, this electromagnetic field is you can think of it as that goes circles around the earth. And that you can think of as as some sort of uh, space of information or whatever. So when you die, you might have you seen any car accident commercials?
Car accident commercials?
And not commercials for car accidents, commercials for like tires or commercials for I think usually commercials for tires in which the car almost hits another car. Okay. Somebody comes out of the blue, and this guy hits, notices at the last moment and hits the brake, and and the next in the next cut, his entire life flashes in front of him. But of course, the tires are good, and he doesn't die. The idea is that at the moment of your death, your entire life experience will be rewind. And that is, I think it's been well documented in empirically in a lot of cases that there is a rewinding of your life experiences at the moment of death. So, and there is also right now, because of the ions flowing and currents flowing in my brain, there is an electromagnetic field. But at the time of moment of death, there is a increase in energy. If you look at the spectrum, so there is a burst of electromagnetic field. So basically, all your life experiences get stamped onto the this global, the planetary electromagnetic field that is going around the Earth. So even you, so now the idea of so then this the electromagnetic field, which you can think of it as the entire life experiences summed together, sum of totality of your life experiences. They get stamped onto this, they get added, or however you do the addition, you might conceive of that, added on to this field. And once it's in the field, it will be there, maybe in some degradation will be there, and then it will be spinning around. And then the idea of rebirth is, under certain conditions, the part of this field gets implanted into the fetus or into the womb, and that's a sketch. That's one of the reasons that, and even the most rational of all religions, in which you can consider Buddhism, even they don't let go of. They say there is no soul, but they won't let go of rebirth.
Oh, okay. Self-contradictory subject. I mean, paradoxical, I would say, instead of self-contradictory.
But I was a little bit surprised that the electromagnetic waves traveling in our brain are aligned with the planetary electromagnetic field, traveling field. That was a little bit disturbing. And when I found
Sorry
out
to
that,
disturb you.
huh?
Sorry to disturb you.
No, it was, but then it was even more disturbing was that there is this, during the moment of death, there is this burst of electromagnetic energy. And that is even more disturbing. Any more questions? Oh, I thought you guys are going to say something. I don't have. Okay, yeah. See, I hope you don't mind, Prashant, if I read what you wrote.
Yeah, he just wrote for us to read out.
No, I was just saying that I'm still reading. I don't have anything to add to that, what I was saying yesterday, the topic.
Yeah.
I'm still doing the reading that you told me and the videos and everything. I'm still working on that.
Yeah, that's fine.
Apart from that, yeah, no, I'm more interested in how, like, what is the data that, that we can take from this and apply it. So I'm still thinking. Yeah, that's all that's there.
Okay.
No reflection as such on today's work, because I'm still like not understanding it fully, actually. To be very honest. Like, it'll take time for me. I need to read it again once, reviewed once, then maybe it'll go in my head. Right now, it's a bit too much for me. Information wise.
Yeah. Yes, I see. I can't even sit in a 1 hour lecture also. Huh? Like for me, all learning is only book learning, and that too I lie on my bed and read most of the time. So I'm really grateful to all of you for listening to me. Sorry. So you see, the reason I'm what is this bugging you to say something is because I think I shared you that book review, The Power of Us. Did I share you that book review?
I don't know.
Let me see.
Let me also check.
I can check. 1 second. Huh? What is this? So what I found really fascinating is that, yeah, especially going back, 1 second. I'll. So, Professor Jagriti said she's going to order that book. The title of the book is Power of Us. So, he brings out, I think, very well, and that there is value. Even if you think of what is this assignment as individual, everybody has to write on their own. If you share and talk and discuss with one another whatever you are trying, I think the final product would be something that would be much more if you were to I just and I think I too subscribe to that idea of see, when you try to explain to somebody or when you try to paraphrase it, or when you try to even simple things like if somebody says Rama killed Ravana, and if you say, oh, Ravana was killed by Rama, then that would also would contribute to improving your understanding. Simply changing the voice passive to active. Sorry. So it is for that reason that it need not be anything, but even rambling, ranting, whatever might be the case, would contribute to some extent in refining. So that is the reason I said loud thinking collective. So collective is probably in the sense of there are all sorts of collectives now. Like I don't know if they still have, but I think when leftists were popular,
I have a question.
Yeah.
Can we write a book review? Like, if we take a book related to neuroscience and then if we read it and write a review, that is also fine, right? For the
If you critique somebody, that is fine too. Somebody's work or somebody's paper. So, see, I would personally think, what is this? More than anything else, I want you to internalize that format that that I shared earlier. You have that format now.
Yeah.
So even the book review, can be written in that format. No, what is this? Say, for example, the power of us. So you can look at that book review. That book review is also very good, and uh, and uh, and there is a subfolder called writing well in the folder that I think I shared that as separate link. There are a couple of articles by Tom Albright. He writes very well. And so a lot of times you have
Last sentence. Can you repeat the name of the author?
Thomas Albright. He is also at Salk. Albright, Albright. They are in the folder that I shared. What is that?
Yeah, got it. The papers et al. Folder.
So Papers et al. Has another folder, writing well. In that, these are all very short papers. So most of these are about some other paper. Like a lot of times, this nature, science, and PNS. I think I mentioned the other day that when they have something very exciting finding, they will ask experts in that field to write a commentary or news on this paper. So it's very important to be able to talk about other people's work also. Book review or reviewing a paper, anything, or a body of literature, they are all fine.
But the ultimate goal is to stick to the format that you have given and then write a paper.
Yeah.
Get the hang of how to write a paper.
You see, the format is simply, all it says is start with something that everybody could understand. Then says, and then slowly. First, you start with terminology or conceptual repertoire that is universally accessible. Say, for example, if I want to, you remember, say for I could say, what is this? Magnesium ion is an open channel blocker of NMDA receptors. So, but the same thing that would be understood by a neuroscientist. But if I want to say convey the gist of that to non-neuroscientists, I would say something like, oh, magnesium ion, NMDA receptor is like a freeway. These ions, calcium and sodium, are like cars going on the freeway, and now all of a sudden a herd of cows wandered onto the freeway, highway, and since they are moving very slowly, and the whole traffic slowed down to a grinding halt. So in the second version, there is something that everybody can understand. So there is very little technical terminology, that insider terminology that I'm using. So that is how you start, and that is how you end. So gradually you can go more and more specialized until you reach a peak where you sum up in specialization means in very definitive concrete terminology, and then you present it in more and more intuitively accessible terminology, and that is how you conclude. So that is the format that um, uh, 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 does it make sense?
Yeah, yeah. So, but the thing is, what I didn't understand is that how can we make it neuroscience and UX? If we, even though if we critique a particular work, we wouldn't know where to look for the interconnection of neuroscience and UX, which is the course title.
Yeah. So. Say, say for example, if you want to write a book review, and then this there is when brains meet buildings. Michael Arbib. I think it's in the drive. It is the title is Brains underscore Buildings. So that one has both neuroscience and UX. So if you want to write a book review, you can write a book review, write a review on that. Can you find it in the papers at all?
Yeah.
Huh?
Yeah, found it, but it is slightly aimed at architectural domain.
Yeah.
I was looking at something that can directly relate to UX. I mean, not experience per se, but like screen design. How does neuroscience directly affect screen design, maybe, or in testing or in designing phase of an application, like a digital application. If there are any papers like that, it will be helpful for us, because this will be a new domain for us altogether. I mean, we have to study.
So like the mobile screen design,
Yeah, it
or
could
the
be
app.
mobile
So
screens.
when you enter, when you
Mobile,
huh?
mobile screens, or it could be products, like digital products or tangible products, so that it is much more we can relate to it quickly, because it's a 1 week module, and too much deviation would be a little difficult for us.
Yeah. So you want to. So what is the user experience that you are hoping to, like when he say for example, if I look at the monitor.
Yeah.
So, I was. Is there any particular user experience that you want the screen, whatever screen you might have in mind, to elicit?
I didn't understand the question. Yeah, every screen has an intent. For example, it is also to evoke a certain experience. So is there any work that is done in this domain so that we can like take off from there? Architecture will be a little difficult for us because, like, at least for me, because I come from very UI UX background.
Basically, what is this? Uh, you can what is this? I could probably what is this? One way to look at it is a screen is some space, and looking at it or would be like navigating that space. I might look, and one thing that I could think of is how would if that space has to is required to house certain number of objects of certain shapes. So, how would you place the objects in that space so that the objects that is required can be readily accessed, or it won't obscure other objects or occlude other objects that are maybe required. So basically, in some sense, there is some sort of optimization involved. So you can drop parallel, or at least you can see it like how are you going to design your room. So you can imagine your room as a screen. So where are you going to place one thing versus another thing? So now with the screen as your room, like say for example, just as one goes around deciding where to put the table or where to put the chair or where to put the table lamp. Isn't the problem that you are talking in terms of screen analogous to this problem of um, uh, putting things in or arranging things in some space in general?
Yeah, the correlation is right, but where what I'm trying to understand is the neuroscience part of it. So there, the space and the digital screen have a correlation. That is not the question. The question is where do I apply neuroscience exactly when I design a particular digital experience.
So one of the things is one could look at it that could inform you. So when you put things, whether in your room or on a screen or design screen. So one of the things that issues constraints or consideration would be what is the thing that you want somebody looking at the screen to see, or somebody who enters your room to see. That might be one consideration, I'm guessing.
Yeah.
So, in other words, there I think the neuroscience that's that talks about how how we look at the world. Like you remember, and the saccadic movements that we started with earlier. So basically, uh, 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 you can make things that you want people to see more salient than the other. So one is I think there is probably a chapter in what is this principles of neural science. I think I named it neural science. Uh, what is this? And there is also uh, my uh, one second. Yeah. 1 second.
You can share it with us tomorrow also, sir, over the mail. I think
No. So you can write. She did work on how. One second. How? Uh, uh, So she does work on learning where to look for basically like uh, how do people go around looking for things. So I think there is this paper title.
Yeah, this looks closer. Yeah.
I think this and this paper, learning where to look. Yeah. Uh, it seems we decide more than one, 170,000 times per day. So maybe if I find additional literature that might inform you, or you can write to her. But when you write to her, say what is this Posina? Otherwise, we used to be colleagues at Salk. So yeah, I think it looks like she's now into what is this? Uh, 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 yeah, I forgot her in the afternoon. What is this? We are usually referred to as neurotypical, and she's also developing this game-based tools to help neurodiverse individuals and I forgot earlier in the afternoons. Somebody, I'm sorry, I forgot your name, but you're talking about this working or developing something for autistic or neurodiverse individuals.
I think that was Neha.
Who is that?
I think
Autistic. No, somebody came here this afternoon. Maybe she said
she is Neha. Neha is the one who asked you. Yes.
Yeah.
She is gone here.
Yeah, I think yeah, she's a very helpful person. Huh?
Okay, I'll read her article. I think her experience also kind of is in the domain of AR, VR, and neuroscience. I think it will be helpful for us. Thanks for the context.
Neha, you remember you came, you wanted to work with neurodiverse individuals. Huh?
Yeah.
Yeah, she did work with artists. I shared the links in the chat. Huh?
Okay.
We can talk again when you come here. Huh?
Okay.
Yeah. Yeah, in a sense, it is about how do you organize things with respect to one or other criteria. So one would be you want something to be readily found, or you want everything to be equally accessible. So there are a range of issues that can come into play, and in general, so the question in the work on attention, I think is something that is probably uh, uh, relevant in the case of designing screens, to simplistically stating. Any more thoughts? Shall we call it a day?
Yes, sir. Yes, sir.
You're waiting for this to end. This talk, is it? Okay, so tomorrow, I think we'll talk about empathy and all sorts of things. Yeah. Is anyone? So I'll see you all tomorrow.
Yes, sir. Yeah.
Yes, sir.
Yes.
Yeah.
Yes, sir.
So, Neha, you're coming down now?
Yes, sir.
Okay.
If you're in a hurry, that's fine.
No, I'm not in a hurry.
Okay.
Sir, Gina here. I had,
Yeah.
I had mailed you, sir, regarding the recording. So what happened is I'm having bad network here. So I was not able to follow few classes also because the video kept breaking up.
Yeah.
So what I will do is I will watch, if you could share me all the recordings, I will go through them. And I'm actually coming to Bangalore soon because attending classes from here is not working for me.
Yeah.
So I'm coming over there, and maybe if I have some doubts, I'll meet up with you and discuss with you.
Yeah. So I see. I'm. What is this? I, he sent me a link. Nibu.
You have shared day one with us. So I have day one. I was able to watch day one video, but day two, day three, and today's I might need.
Yeah, absolutely. But when did he send that? Let me see. So
That's day 1, 13. Yeah.
So the one, it doesn't let you see any other days, huh? Let me see. Passcode. Oh, it is asking for some password, huh?
No, you can play it now. I think the passcode you just entered, right? Right now the dialog box says to save password. I don't think that matters.
Save password, I'll say.
If you play if you play this video, this is day 1. But I'm not sure if we can get day 2 and day 3 with the same link. I don't think we can.
But this one is playing, or do we have to do anything? Huh?
Just click on that play button. It will play. No?
Product. So in order to understand, in order to get a better handle of the product, it is useful to understand what is the process that gave rise to it. Simply, this is here. Stop. I have. Here it says download 3 files. I was wondering maybe the 3 files correspond to. Do you know anything about Zoom?
I'm also not aware. Let me also check it from my end, sir. Let me see. I only thought I didn't see the 3 files thing.
The 3 files are on the what is this? Right hand side, top right hand corner. It said download 3 files. Right. No, is it downloading or no? No. What is this?
So those 3 files are of the same day. One will be a text file of the chat messages. The other will be like the other 2 will be the videos in 2 different formats.
Okay. So then I'll ask him. Do you know where this chat is getting saved?
So the recordings have the chat. Like if we are getting all the recordings, you will get all the chat also.
Oh, okay.
This computer will check whether this one. This is the chat. So it's saving here.
No, yesterday, 9:15.
So you want today's? This one is today's. It's in documents. So I'll just transfer this to your file, Raidu file.
But you rename it.
I'll rename it. Okay, I need to rename
It's already.
Yes. Then documents. So 16th, right?
Just. The recordings are not there, no? Nibu send this link.
Yeah.
Only one recording is there, no?
Yeah.
Other recordings are not there. So I have to ask him.
It will be saved in the cloud actually. So he will have everything. So,
but the cloud link that he shared doesn't have.
Doesn't have everything.
Okay, so I'll send an email to. Do you guys want to go get some coffee or tea? I do. Yeah. No, it's okay. Huh? Oh, it's okay. It's all right. You don't feel like having tea? You don't feel like having tea? Then we all can go.